The following is a conversation with Betül Kaçar, an astrobiologist at University of Wisconsin, studying the essential biological attributes of life. And now, a quick few-second mention of each sponsor. Check them out in the description. It's the best way to support this podcast. We got House of Macadamias for a satiating and delicious snack, Mizzenamain for style, 8 Sleep for naps, ExpressVPN for privacy and security, Give Directly for philanthropy, and Blinkist for nonfiction. Choose wisely, my friends. And now, onto the full ad reads. As always, no ads in the middle. I try to make this interesting, but if you skip them, please still check out our sponsors. I enjoy their stuff. Maybe you will too. This show is brought to you by House of Macadamia. It's a company that ships delicious, high-quality, and healthy macadamia nuts directly to your door, which they did just today. Another shipment. I got in my hand an unopened box, dipped macadamia's chocolate flavor. This is the one I went through first last time, and it was delicious. It is an incredible source of happiness for me. I knew for a long time that macadamia nuts were really healthy, because I remember when I was first exploring, okay, so on keto, what is the way to get fats into the diet? Because it has to be a high-fat diet. And I was looking at nuts and I was thinking of it, okay, which nuts are the healthiest? And I remember macadamia is always coming up to the top of the list. I'm going to try not to eat all of them so that I have some for guests when they come over, because it's like a perfect guest snack. It's healthy, it's delicious, it's just pure perfection. They also sent me some macadamia bars, which were also delicious last time. I probably prefer the whole nuts. I'm nuts for nuts. Anyway, go to houseofmacadamias.com/Lex to get 20% off your first order. This show is also brought to you by Mizzenamain, the maker of comfortable, stylish dress shirts and other menswear. I wear their black dress shirt, and I love it. I have a ton of them. Sometimes when I do these ad reads, I struggle to try to convey exactly why I like this particular piece of clothing, which is where I wish there was like a brain-computer interface way to directly communicate the actual experience of wearing the clothing. And if you get a large number of people with that data, you get a sense of, okay, well, 96% of people feel good in this. That's a good chance that I will also feel good in. That's fascinating. Of course, you could get that through self-report surveys, but it would be nice to get direct data because you can't lie, but the feelings don't lie. But then again, there could be other kinds of variables, like what else is going on in your life. You might be just in a bad mood about something else, or watch a really sad movie while wearing the shirt. So I don't know. It's an imperfect system that we're going to have to work out. Right now, you'll get $35 off any regular price order when you go to mizzenamain.com and use promo code LEX. This episode is also brought to you by 8 Sleep and its new Pod 3 mattress. They've been a sponsor forever. And they've been my bed mattress cooling system when I sleep forever. And it's a source of a lot of happiness. I recently tweeted about the number of hours there are in a regular lifetime. You know, if you live 70 to 80 years old, there's about 600 to 700,000 hours in a life. And I think a large percent of those hours is spent sleeping. Now, of course, you can think of sleeping as an important process for health to regenerate your energy and all that kind of stuff. But it is also in itself a source of happiness. And I think you want to use the best bed for the job. And there's just not a better feeling of a cool surface with a warm blanket. And a day full of battles won. And you return to rest and reminisce about the battles. So you can again, the next day, return to the battles that are in store for you. Check it out and get special savings when you go to asleep.com/Lex. This show is also brought to you by ExpressVPN. I've used them for many years. They've had the big, sexy button that just works when you press it. And I use it, it's the first layer of protection of your data, of your privacy on the internet, ISPs, and basically a lot of different services and tools collect your data. And a good VPN should be your first layer of protection in those situations, in most situations. You can also change your location in the world. Now, this is useful for different services like Netflix and so on. But mostly, I just like it from a software engineering design perspective. I love tools that just work. They do one thing and they do it really well. And they do it across different platforms. They never break down. Everything just works. Everything's intuitive from the UI perspective. And from the backend perspective, everything just works smoothly. Android, Linux, everything. I love it. Go to expressvpn.com/LexPod for an extra 3 months free. This show is also brought to you by Give Directly, a nonprofit that lets you send cash directly to people in need. It's perhaps a counterintuitive notion, but the giving of money to alleviate some of the suffering in the world or to improve the quality of life for a bunch of people in the world is not an easy problem. I mean, there's so many challenges, and there's just a lot of studies behind the principles that Give Directly operates under, which is if you actually give money to the people most in need, they're going to themselves spend that money to maximally improve their life. This too is a counterintuitive notion, but it works time and time again. Like many things, I have so much to learn in this world about challenging problems like this. So philanthropy is definitely one of them. So I should also mention that a bunch of previous guests of this podcast have used Give Directly to donate money. Jack Dorsey, Elon Musk, Vitalik Buterin, and several others. Visit givedirectly.org/Lex to learn more and send cash directly to someone living in extreme poverty. That's givedirectly.org/Lex. This show is also brought to you by Blinkist, my favorite app for learning new things. Blinkist takes key ideas from thousands of nonfiction books and condenses them down into 15 minutes that you can read or listen to. I set the goal of reading one book a week. I said a classic book, but really what I mean is a book that really challenges you and really makes you think. So I look forward to seeing sort of using Blinkist to help me review the books, actually also choose the books, and maybe help others that are interested in following along with my reading to get deeper and deeper insights into these different books. I think my perspectives on many books I read are non-standard. I get lost on a single quote here and there, and it just kind of takes me down this whole tangent of thought, these ideas that get inspired and get sparked, and then I just construct this whole world from a single quote sometimes. But that's what makes it interesting. It's a very subjective experience, like appreciating art, appreciating books, and so on. Anyway, you can claim a special offer for savings at Blinkist.com/Lex. This is the Lex Friedman Podcast. To support it, please check out our sponsors in the description. And now, dear friends, here's Betül, Kaçar. What is the phylogenetic tree or the evolutionary tree of life? And what can we learn by running it back and studying ancient gene sequences as you have?
I think phylogenetic trees could be one of the most romantic and beautiful notions that can come out of biology. It shows us a way to depict the connectedness of life and all living beings with one another. It itself is an ever-evolving notion. Biologists like visualizations. They like these graphics, these diagrams, and tree of life is one of them.
So the tree starts at a common ancestor.
It's actually the other way around. It starts from
At the end?
It starts from the branches. It starts from the tip of the branch, actually, and then the further, depending on what you collected to build the tree. So depending on the branches, depending on what's on the tip of the branch, and I will explain what I mean, the root will be determined by what is really sitting on the tip of the branch of the tree.
So we could study the leaves of the tree by looking at what we have today and then start to reverse engineer, start to move back in time to try to understand what the rest of the tree, what the roots of the tree look like.
Exactly. So the tree itself, by just taking a few steps back and looking at the entire tree itself, can give you an idea about the connectedness, the relatedness of the organisms, or whatever, again, you use to create your tree. There are different ways. But in this case, I'm imagining entire diversity of life today is sitting on the tips of the branches of this tree. And we look at biologists, look at the tree itself. We like to think of it as the topology of the tree to understand when certain organisms or their ancestry may have merged over time. And depending on the tools you use, you might use this tree to then reconstruct the ancestors as well.
And so, what are the different ways to do the reconstruction? So you can do that at the gene level, or you could do it at the higher complex biology level, right? So, in which way have you approached this fascinating problem?
We approached it in every way we can. So it's the gene, could be protein, the product of the gene, or species, or could be even groups of species. It will depend. It totally depends on what you want to do with your tree. If you want to understand certain past events, whether an organism exchanged a certain DNA with another one along the course of evolution, you can build your tree accordingly. If you rather use the tree to reconstruct or resurrect ancient DNA, which is what we do, then in our case, for instance, we do both gene, protein, and species because we want to compare the tree that we create using these different information.
Okay, well, let me ask you the ridiculous question then. So, how realistic is Jurassic Park? Can we study the genes of ancient organisms, and can we bring those ancient organisms back? So, the reason I asked that kind of ridiculous sounding question is maybe it gives us context of what we can and can't do by looking back in time.
Yeah. So, dinosaurs or all these mammals in at least for us is the exciting thing already happened by the time we hit to larger organisms or to eukaryotes.
Oh, to you, the fun stuff is before we got to the mammals.
The fun stuff is what people think is boring, I think. The phase that, well, at least two different times in the geologic history. One is the first life, past origin of life. How did first life look like? And the second is, why do we think that over certain periods of geologic time, no significant innovation happened to the degree of leaving no record behind?
So what do we not have a record of? Which part? So you say the fun stuff to you is after the origin of life, which we'll talk about. After the origin of life, there's single cell organisms. The whole thing with the photosynthesis, the whole thing with the eukaryotes and multicell organisms, and what else is the fun stuff? The whole oxygen thing, which mixes in with the origin of life. There's a bunch of different inventions, all that have to do with this primitive kind of looking organisms that we don't have a good record of.
So I will tell you the more interesting things for us.
Sure.
One is the origin of life, or what happened right following the emergence of life. How did the first cells look like? And then pretty much anything that we think shaped the environment and was shaped by the environment in a way that it impacted the entire planet, that enabled you and I to have this conversation. We have very little understanding of the biological innovations that took place in the past of this planet. We work with a very limited set of, I don't want to even say data because they're fossil records. So let's say imprints, either that comes from the rock and the rock record itself, or what I just described, these trees that we create and whatever we can infer about the past. So we have two distinct ways that comes from geology and biology, and they each have their limitations.
Okay, so, right, so there's an interplay. The geology gives you that little bit of data, and then the biology gives you that little bit of kind of constraints in the materials you get to work with to infer how does this result in the kind of data that we're seeing. And now we can have this through the fog. We can look back hundreds of millions of years, a couple of billion years, and try to infer.
Even further, and I like that you said fog. It is pretty foggy. And it gets foggier and foggier the further you try to see into the past. Biology is you basically study the survivors, broadly speaking, and you're trying to put together their history based on whatever you can recover today. what makes biology fascinating also it erased its own history in a way, right? So you work with this 4 billion year product, that's genome, that's the DNA. It's great. It's a very dynamic, ever evolving chemical thing.
Mhm.
And so you will get some information, but you're not going to get much unless you know where to look, um, because it is responding to the environment.
Yeah, so what we have, that's fascinating. What we have is the survivors, the successful organisms, even the primitive ones, even the bacteria we have today.
So bacteria is not primitive, and we
Sorry to offend the bacteria.
we should be very grateful to bacteria. First of all, they are our great ancestors. I like this quote by Douglas Adams: "Humans don't like their ancestors; they rarely invite them over for dinner."
Yeah.
Right. But bacteria is in your dinner. Bacteria is in your gut. Bacteria is helping you
So we do
along the
invite
way.
them for dinner.
We invite them. Well, they get themselves invited in a way.
Yes.
And so and they're definitely older, and definitely very sophisticated, very resilient than anything else. As someone working as a bacteriologist, I feel like I need to defend them in this case because they don't get much shout out. When we think about life.
So you do study bacteria. So which organisms gives you hints that are alive today that give you hints about what ancient organisms were like? Is it bacteria? Is it viruses? What do you study in the lab?
We study a variety of different bacteria depending on the questions that we ask. We engineer bacteria. So ideally, we want to work with bacteria that we can engineer. Seldom we developed the tools to engineer them. And it depends on the question that we are interested in. If we are interested in connecting the biology and geology to understand the early life and fundamental innovations across billions of years, there are really good candidates like cyanobacteria. So we use cyanobacteria very frequently in the lab. We can engineer its genome. We can perturb its function by poking its own DNA with the foreign DNA that we engineer in the lab. We work with E. coli. It's the most simple in terms of model systems goes. Organism that one can study, well established, sort of a lab pet that we use it a lot for cloning and for understanding basic functions of the cell, given that it's really well studied.
So, and what you do with that E. coli, you said that you inject it with foreign DNA?
We inject pretty much all the bacteria that we work with foreign DNA. We also work with diazotrophs. These are azotobacteria. They're one of the prime nitrogen fixers, nitrogen fixing bacteria.
Can you explain what that is? Nitrogen fixing? Is the source of its energy?
So nitrogen is a triple bond gas that's pretty abundant in the atmosphere, but nitrogen itself cannot be directly utilized by cells given it is triple bond. It needs to be converted to ammonia that is then used for the downstream cellular functions.
And that's what counts as nitrogen fixing.
Yes, so nitrogen needs to be fixed before our cells can make use of it. And it's
no offense to nitrogen either.
Well, it's actually a very important element. It's one of the most abundant elements on our planet that is used by biology. It's in ATP. It's in chlorophyll. That relies on nitrogen. So it's a very important enzyme for a lot of cell functions.
And there's just one mechanism that evolution invented to convert it, fix it?
So far we know there's only one nitrogen fixation pathway, as opposed to, say, carbon. You can find up to 7 or 8 different carbon base microbes invented to fix carbon. That's not the case for nitrogen. It's a singularity across geologic time. We think it evolved around 2.7, maybe roughly 3, probably less than 3 billion years ago. And that's the only way that nature invented to fix the nitrogen in the atmosphere for the subsequent use.
Would we still have life as we know it today if we didn't invent that nitrogen fixing step?
I cannot think of it, no. It's essential to life as we know. You and I are having this conversation because life found a way to fix nitrogen.
Is that one of the tougher ones? If you put it sort of oxygen, nitrogen, carbon, what are, in terms of being able to work with these elements, what is the hardest thing? What is the most essential for life? Just to give context.
Well, we think of this as the cocktail. You may hear
what's in the cocktail?
It's the schnapps, right? Carbon, hydrogen, oxygen, nitrogen, sulfur. So there are 5 elements that life relies on. We don't quite know whether that's the only out of many options that life necessarily needs to operate on, but that's just how it happened on our own planet. Mm-hmm. And there are many abiotic ways to fix nitrogen, and like lightning, right? Lightning can accumulate ammonia. Humans found a way about 100 years ago, I think around World War I, the Haber-Bosch process, that we can abiotically convert nitrogen into ammonia. Actually, 50% of the nitrogen in our bodies comes from the human conversion of nitrogen to ammonia. It's helped. It's the fertilizer that we use, urea, comes from that process. It's in our food. So we found a way to fix our own nitrogen for ourselves.
Yeah, but that's way after the original invention of how to fix nitrogen.
Absolutely.
And without that, we wouldn't have all the steps of evolution along the way.
Oh, absolutely. We tried to replicate in the most simplest way what nature has come up with, right? We do this by taking nitrogen, using a lot of pressure, and then generating ammonia. Life does this in a more sophisticated way, relying on one single enzyme called nitrogenase. It's the nitrogen that is used together with 8 electron donor and ATP, together with a lot of hydrogen. Life pushes this metabolism down to create fixed nitrogen. It's quite remarkable.
So the lab pet, E. coli, inject them with DNA. Those are so E. coli does nitrogen fixing in part, or was that a different one?
So some biological engineers engineered E. coli to fix nitrogen, I believe. Not us. We use the nature's nitrogen fixing bug and engineer it with the nitrogen fixing metabolism that we resurrected using our computational and phylogenetic tools.
How complicated are these little organisms? What are we talking about?
It depends on how we define complication.
Okay, so I could tell that you appreciate and respect the full complexity of even the most seemingly primitive organisms, because none of them are primitive. Okay. That said, what are we talking about? What kind of machineries do they have that you're working with when you're injecting them with DNA?
So I will start with one of the most fascinating machineries that we target, which is the translation machinery. Mm-hmm. It is a very unique subsystem of cellular life in comparison to, I would say, metabolism. And we used to, when we are thinking about cellular life, we think of cell as the basic unit or the building block. But from a key perspective, that's not the case. That one may argue that everything that happens inside the cell serves the translation and the translation machinery. There's a nice paper that called this that the entire cell is hopelessly addicted to this main informatic, computing, biological, chemical system that it is operating at the heart of the cell.
Which is the translation?
It is the translation.
What's translation from what to what? So RNA to enzymes?
It converts a linear sequence of mRNA into a later folded protein. That's when that's the core processing center for information for life. It has multiple steps. It initiates, it elongates, it terminates, and it recycles. It operates discrete bits of information. Itself is like a chemical decoding device. And that is incredibly unique for translation that I don't think you will find anywhere else in the cell that does this.
So even though it's called translation, it's really like a factory that reads the schematic and builds a three-dimensional object. It's like a printer.
I would divide it into actually even 4 more additional steps or disciplines than what would it take to study it by the way you described it. It's a chemical system. It's the compounds that make it up are chemicals. It's physical. It tracks the energy to make its job, to do its job. It's informatic. What is processed are the bits. It's computational. The discrete states that the system is placed when the information is being processed. That's itself is computational, and it's biological. It's there's variability and inheritance that come from imperfect replication, even and imperfect computation. So
Man, that's so good. So from the biology comes the like when you mess up, the bugs are the features. That's the biology. Informatics is obvious in the RNA. That's a set of information there. The different steps along the way is actually kind of what the computer does with bits. It's done computation. Physical, there's I guess like almost like a mechanical process to the whole thing that requires energy, and actually, it's manipulating actual physical objects. And chemical is because ultimately it's all chemistry.
Yeah, and it tracks this information. So it is almost a mini computer device inside our cells.
Yeah.
And that's the oldest computational device of life. It's likely the key operation system that had to evolve for life to emerge.
It's more interesting, or it's more complicated in interesting ways than the computers we have today. I mean, everything you said, which is really, really nice. I mean, I guess our computers have the informatic and they have the computational, but they don't have the chemical, the physical, or the biology.
Exactly. And the computers don't link information to function, right? They are not tightly coupled, nowhere close to what translation or the way translation does it. So that's the number one, I think, difference between the two. And yes, it's informatic, and we can discuss this further too.
100%. Let's please discuss this further. Which part are we discussing further? Each one of those are fascinating worlds. Each of the 5.
Yeah. So we can start with the more, I guess, the ones that are more established, which is the chemical aspect of the translation machinery. It's the specific compounds make up the assembly of RNA. Chemists showed this in many different ways. We can rip apart the entire machinery. We know that at the core of it, there's an RNA that operates not only as an information system itself or information itself, but also as an enzyme. And origin of life chemists make these molecules easily now. We know we can manipulate RNA. We can make, even with single pot chemistries, we can create compounds.
What's a single pot chemistry?
That's I would say when you add all the recipes that you know that will lead you to the final product.
This is what original life chemists do. Is they come up with this pot, they throw a bunch of chemicals in, and they try to they're basically chefs of a certain kind.
I'm not sure if that's what they call it, but that's how I think of it, because it is all combined in a test tube, and you know the outcome, and it's very mathematical once you know the right environment and the right chemistry that needs to get into this container or this pot. You know what the outcome is. There's no luck there anymore. It's a pretty rigid, established input-output system, and it's all chemistry.
So you actually wear a lot of hats. Is one of them origin of life chemist?
My PhD is in chemistry, but I don't do origin of life chemistry.
But you're interested in origin of life.
Yes, absolutely.
So some of your best friends are origin of life chemists.
Just make sure that you have good chemist friends if you're interested in origin of life.
Yeah,
that's 100% requirement. It should be mandatory.
Okay, so chemistry. So what else about this machinery that we need to know chemically?
Well, chemically, I think that's it. You have enzymes, you have proteins. The enzymes are doing their thing. They know how to chew energy using ATP or GTP. They know what to do in their own way. They do their enzymatic thing. So it's not just the ribosome that is at the heart of the translation, but there are a lot of different proteins. You're looking about 100 different components that compose this machinery.
Well, let me ask maybe it's a ridiculous question, but did the chemistry make this machine, or did the machine use chemistry to achieve a purpose? So I guess there's a lot of different chemical possibilities on Earth. Is this translation machinery just picking and choosing different chemical reactions that it can use to achieve a purpose? Or did the chemistry basically there's a momentum, like a constraint to the thing that can only build a certain kind of machinery? There's basically is chemistry fundamental, or is it just emergent? How important is chemistry to this whole process? I guess
you cannot have life without chemistry. You cannot have any cellular process without chemistry. What makes life interesting is that even if the chemistry is imperfect, even if there are accidents along the way, if something binds to another chemical in a way it shouldn't, there is resilience within the system that it can maybe not necessarily repair itself, but it moves on. However imperfect, mistakes can be handled.
That's where the biology
comes in. But in terms of chemistry, you absolutely cannot have a translation machinery without chemistry. And so you're, as I said, there are 4 main steps. These are the core steps that are conserved in all translation machinery. And I should say, all life has this machine, right? Every cell, everything.
On Earth.
On Earth.
Yeah.
Yes.
When you think of this machine, do you think very specifically about the kind of machinery that we're talking about, or do you think more philosophically, a machine that converts information into function?
I cannot separate the two. I think what makes this machinery fascinating is that those 5 components that I listed they coexist. So, for instance, if we let's just talking about the chemistry part, we know the certain rate constant all these proteins that operate in this machinery needs to harbor in order to get the mechanism going, right? If you are bringing the information to the translation machinery and you are the initiator of this computation system, you need to have you can only afford a certain range of mistakes. If you're too fast, then the next message cannot be delivered fast. If you're too slow, then you may stall the process. So there is definitely a chemistry constant going on within the machinery. Again, it's not perfect, far from it, but they all have their own margin of error that they can tolerate versus they cannot. Otherwise, they collapse. The system collapses.
So it's like a jazz ensemble, the notes of the chemistry, but you can be
I
a
like
little
that
offbeat.
You said jazz. It's definitely true. It's a party, and it's like everybody's invited, and they need to operate together, right? And they what's really cool about it, I think, or there are many things that are very interesting about this thing. But if you remove it from the cell and put it in a cell-free environment, it works just fine, right? So you can get cell-free translation systems. Put this translation in a test tube, and it is doing its thing. It doesn't need the rest of the cell to translate information. Of course, you need to feed the information, at least so far. But because we are far from evolving a translation, maybe not so far, evolving a translation in the lab or a machinery that can process information as it generates it. We have not done that yet.
It's a pretty complicated machinery, so it's hard for those origin of life chemists to find a pot that generates.
Because it's far more than chemistry. You need biology, obviously. You need biochemistry. You need to think as a network systems folk. You need to think about computation. You need to think about information, and that is not happening yet. Except we are trying to bring this perspective. But the more you understand how information systems work, you cannot, once you see it, you cannot unsee it. It's one of those things.
So, but you can still rip it out and the chemistry happens.
Yes. And chemistry can happen even with, even if you strip some of the parts out. It can, you can get very minimal level of information processing that does not look anything like the translation that cells relies on, but chemists showed from linear, you can generate information that arrives to a processing center in the form of a linear polymer. But the informatic part of this system that I think sets it apart from computation and from metabolism comes in if you think about the information itself, right? So we have 4 nucleotide letters that compose DNA, and they are processed in the translation in triplets. So you have in triplet codon fragments. So you have 4 times 4 times 4. So you have 64 possible states that can be encoded by 4 letters in 3 positions.
Mm-hmm.
All right. So
it's so amazing.
Yeah.
It's so amazing.
There is only one code that says start. That there's only one. And then there's 2, if not 3, that says stop. So that's what you work with. But you can have 64 possible states, but life only uses 20 amino acids. So life uses 64 possible states minus 4 of the starts and stops to code for 20 amino acids in different combinations. That is really amazing. If you think about there are 500 different amino acids life can choose, right? It's narrowed it down to 20. We don't know why. A lot of people think about this genetic code is quite fascinating
so far,
in its own right. I mean, it didn't do it for 4 billion years. I don't know. We may wait for another 4 billion years, but
you didn't have those amino acids in the very beginning, right? Like
we don't know. So we would be fooling ourselves if we said we know exactly how many amino acids existed early on. But there's no reason to think that it wasn't the same
or
set.
Similar.
Yeah, we don't have a good reason, but because roughly 20 out of 60 states are used, you're using 1/3 of your possible states in your information system. So this may seem like a waste, but informatically it's important because it is abundant and it is redundant, right?
Yeah.
So this code degeneracy, you see this that's implemented by this translation machinery inside the cell. So it means you can make errors, right? You can make errors, but the message will still get through. You can speak missing some letters, to the information can miss some parts, but the message will still get through. So that 2/3 of the not used states gives you that robustness and resilience within the system.
So at the informatic level, there's room for error. There's probably room for error in all 5 categories that we're talking about. There's probably room for error in the computation. There's probably room for error in the physical.
Yes, exactly.
Everywhere there's room for error.
Because this informatic capacity is made possible together with the other components. And not only that, but also the product yields a functional, in this case, enzyme or protein, right? So that's really amazing to me.
It is. I mean, in my head, just so you know, because I'm a computer science AI person, the parallels between even like language models that encode language, or now they're able to encode basically any kind of thing, including images and actions, all in this kind of way. The parallel in terms of informatic and computation is just incredible.
Actually, I have a image maybe I can send you.
Can we pull it up now?
If you just do genetic codon charts, we can pull that up. Yeah, it's a very standard table. So I can explain why this is so amazing. So you're looking at like this is life's alphabet, right? And so I also want to make a very quick link now to your first question, the tree of life. When we try to understand ancient languages, right, or the cultures of the cultures that use these extinct languages, we start with the modern languages, right? So we look at Indo-European languages and try to understand certain words and make trees to understand, this is what Slavic word is for snow, something like snig.
Now we
This
jump
is
to languages that humans spoke throughout
human
human
spoke.
history.
Exactly. So we make trees to understand what is the original ancestor, what did they use to say snow. And if you have a lot of cultures who use the word snow, you can imagine that it was snowy. That's why they needed that word. It's the same thing for biology, right?
Yeah.
If we understand some function about that enzyme, we can understand the environment that they lived in. It's similar in that sense. So now you're looking at the alphabet of life. In this case, it's not 20 or 25 letters. You have 4 letters. So what is really interesting that stands out to me when I look at this, on the outer shell, you're looking at the 20 amino acids that compose life, right? Mm-hmm. The methionine that you see, that's the start. So the start is always the same. To me, that is fascinating, that all life starts with the same start. There's no other start code. So you send the AUG to the cell, that when that information arrives, the translation knows, all right, I got to start, function is coming. Mm-hmm. Following this is a chain of information until the stop code arrives, which are highlighted in black squares.
So for people just listening, we're looking at a standard RNA color table organized in a wheel. There's an outer shell and there's an inner shell, all using the 4 letters that we're talking about. And with that, we can compose all of the amino acids. And there's a start and there's a stop. And presumably you put together with these letters, you walk around the wheel to put together the words, the sentences that
the words, the sentences. And you get one start. You get 3. There are 3 different ways to stop this. One way to start it, and for each letter, you have multiple options. So you say you have a code A, the second code can be another A, and even if you mess that up, you still can rescue yourself. So you can get for instance, I'm looking at the lysine, the K. You get an A, and you get an A, and then you get an A that gives you the lysine, mm-hmm. right? But if you get an A, and if you get an A and then get a G, you still get the lysine. So there are different combinations. So even if there's an error, we don't know if these are selected because they were erroneous and somehow they got locked down. We don't know if there's a mechanism behind this or we certainly don't know this definitively. But this is informatic part of this. And notice that the colors, and in some tables too, the colors will be coded in a way that the type of the nucleotide can be similar chemically. But the point is that you will still end up with the same amino acids or something similar to it, even if you mess up the code.
Do we understand the mechanism, how natural selection interplays with this resilience to error? So like which errors result in the same output, like the same function, and which don't? Which actually results in a dysfunction, or which are...
We understand to some degree how translation and the rest of the cell work together, how an error at the translation level, this is the really core level, can impact entire cell. But we understand very little about the evolutionary mechanisms behind the selection of the system. It's thought to be as one of the hardest problems in biology, and it is still the dark side of biology. Even though it is so essential. So this is, you're looking at the language of life, so to speak, and how it can found ways rather to tolerate its own mistakes.
So the entire phylogenetic tree can be like deconstructed with this wheel of language.
Because all the final letters, that's the 20 amino acids, that's our alphabet. Yeah. They are all brought together with these bits of information, right? So when you look at the genes, you're looking at those 4 letters. When you look at the proteins, you're looking at the 20 amino acids, which may be a little easier way to track the information when we create the tree.
So using this language, we can describe all life that's lived on Earth.
I wish.
One perspective.
We are not that good at it yet, right?
So in theory, this is one way to look at life on Earth.
if you're a biologist and you want to understand how life evolved from a molecular perspective, this would be the way to do it. And this is what nature narrowed its code down to. So when we think of nitrogen, when we think of carbon, when we think of sulfur, it's all in this. That all these nucleotides are built based on those elements.
And this is fundamentally the informatic perspective on this.
Exactly. That's the informatic perspective. And it's important to emphasize that this is not engineered by humans. Mm-hmm. This evolved by itself.
Like, right. Humans didn't invent this just because we're just describing, we're trying to describe the language of life.
It appears to be a highly optimized chemical and information code. It may indicate that a great deal of chemical evolution and this may indicate that a lot of selection pressure and Darwinian evolution happened prior to the rise of last universal common ancestor, because this is almost a bridge that connects the earliest cells to the last universal common ancestor.
Okay, can you describe what the heck you just said? So this mechanism evolved before the what common ancestor?
So there's the
last universal common ancestor.
Yes, so when we talk about the tree, when we think about the root, if you ideally included all the living information or all the available information that comes from living organisms on your tree, then on the root of your tree lies the last universal common ancestor, LUCA, right?
Why last? Last universal. Because the earlier universal, it also had trees, but they all died off.
We call it the last because it is sort of the first one that we can track, because we don't know what we cannot track, right?
There was one organism that started the whole thing.
It's more like I would think of it as more like a population, a group of organisms than a single.
Hold on a second. I tweeted this, so I want to know the accuracy of my tweet. All right. Sometimes early in the morning, I tweet very pothead-like things. I said that we all evolved from one common ancestor that was a single cell organism 3.5 billion years ago, something like this. How true is that tweet? Do I need to delete it? No, there's actually correct. I mean, I think, of course, there's a lot to say, which is like we don't know exactly. But to what degree is that the single organism aspect, is that true versus multiple organisms?
Do you want me to be
brutally honest? Yes, please. This.
So
there's still time. This is how we did like caveats to tweets.
All right. So first of all, it's not 3.5 is still a very conservative estimate. That's just
in which direction?
I would say it's 3.8 is probably safer to say at this point.
A bunch of people said it probably way before.
If you put an approximately, I'll take that.
I didn't. I just love the idea that I was once, first of all, as a single organism, I was once a cell.
Well, you're a group of cells.
No, but I started from a single cell. Me, Lex.
You mean like you versus Luca? Are you relating to Luca right now?
No, I'm relating to my
your
Lex,
own development.
My own development. I started from a single cell. It's like it like built up and stuff. Okay, that. And then, so that's for single biological organism. And then from an evolutionary perspective, the Luca, like I start, like my ancestor is a single cell. And then here I am sitting half asleep, tweeting. Like I started from a single cell, evolved a ton of murder along the way to lead this like brutal search for adaptation through the 3.8 billion years.
So you defy the quote of Douglas Adams. You are proud of your ancestors, and you invite them over to dinner, and you invite them over to your Twitter.
Yeah. And it's amazing that this intelligence, to the degree you can call it intelligence, emerged to be able to tweet whatever the heck I want.
Yes, I mean, it's almost intelligence at the chemical level, and this is also probably one of the first chemically intelligent system that evolved by itself in nature.
Yeah, so you see that translation is a fundamentally like intelligent mechanism.
in its own way, and again, the if we manage to figure out how to drive life's evolution, in it can, if it can evolve a sophisticated sort of informatic um, processing system like this, you may ask yourself what might chemical systems be capable of independently doing under different circumstances.
Yeah, so like locally, they're intelligent locally. They don't need the rest of the shebang. Like they don't need the big
They
picture.
need, so that's a great segue into what makes us biological, right? The heart of the cellular activities are translation. You kill translation, you kill the cell.
Yes.
You not only the translation itself, you kill the component that initiates it, you kill the cell. You remove the component that elongates it, you kill the cell. So there are many different ways to disrupt this machinery. All the parts are important. Now it can vary across different organisms. We see variation between bacteria versus eukaryotes versus archaea, right? So it is not the same exact steps, but it can get more crowded as we get closer to eukaryotes, for instance. But you are still computing about 20 amino acids per second, right? This is what you're generating every second.
That single machinery is doing 20 a second?
21 for bacteria, I believe 8 for eukaryotes, or 9.
21 a second. I mean, that's super inefficient or super efficient, depending on how you think about it.
I think it's great. I mean, I can.
Yeah, but it's way slower than a
acids.
computer could generate through simulation.
I think if you can show me a computer that does this, we are done here.
Well, this is the big. This includes the 5 things, not just. But I could show you a computer that's doing the informatic, right?
Yes, you can show me that, but you cannot show me the one that has all.
For now, I will ask you about probably what AlphaFold, right?
I think the more we learn about, and this is why early life and origin is also very fascinating and applicable to many different disciplines. There's no way you see this the way we just described it unless you think about early life and early geochemistry and earliest emergent systems. But going back to the biological component, all of these attributes that we think about life or that we associate with biology stems from translation and as well as metabolism. But I see metabolism as a way to keep translation going, and translation keeps metabolism going. But translation is arguably a bit more sophisticated process for the reasons that I just described.
So metabolism is a source of energy for this translation process.
It's a way to process materials, and it is inherently dynamic, and it is flexible, but it is not focused on repetition as translation does. So that's the main difference. Translation is the kind of, in a way, just it repeats, right? So you have the metabolism that can synthesize materials, creates or benefits from available energy, and again, it's a dynamic system. And then you have computation that is inherently repetitive, right? Needs to carry out repetitive processes, it does the tasks, and it implements an algorithm, but it is not dynamic. So you see both of those attributes in translation combined. It is repetitive, and it is dynamic, and it also processes this information. So they are fundamentally different. I don't know if you can get life if you don't find a way to process the information around you
in a repetitive, dynamic way.
Yeah, and somehow that's what got selected. Maybe not selected. I don't know if it was accidental, but that's what it seems to be conserved for 4 billion years. That's what life established upon.
What's the connection between translation and the self-replication, which seems to be another weird thing that life just started doing, wanting to just replicate itself?
I think when we truly understand the answer to that question, we may have just made ourselves life, right? I don't think we know quite how translation machinery as a whole fits into the equation, because we try to understand ribosomes, RNA, how the linear information is processed, or the genetic code, why this codons, not others, Y20, not more, not less. And we are sort of moving towards transition. That's what we are working on anyway, to finally look at the patterns in which this system operates itself. And if you understand that, you're really unlocking a very emergent behavior.
One of the things you didn't mention is physical. Is there something to mention about that component that's interesting?
There's actually a paper published in 2013. I want to say the first author is Zirnov. So they surveyed a computational engineered systems level computation energy consumption. Okay. And they tried to understand whether the universe is using its own, or life is using its full capacity of energy consumption, and whether if different planets in the universe had life, would the capacity would increase or decrease. Does life operate at its energy maximum? And they think that it does, that it actually operates at an efficiency that is far more above and beyond any computational system.
How is that possible to determine at all?
That you tell me. That's why I dropped the citation.
Yeah.
I found the citation. It's quite an interesting paper. It's a bit, it's a obviously you can only calculate and infer these things, but
it's a good question to ask. Is the life that we see here on Earth and life elsewhere in the universe, is it the energy most efficiently?
Yeah.
It seems to be very efficient. I mean, again, if we compare to computers, it seems to be incredibly efficient at using energy.
I think they look at the theoretical optimum for electronic devices.
Got it.
And then try to understand where life falls on this, and life is certainly more efficient.
And that's ultimately the physical side. How well are you using for this entire mechanism the energy available to you? And so given all the resilience to errors and all that kind of stuff, it seems that it's close to its maximum.
Yep.
And this paper aside, it does seem that life, obviously, that's the constraint we have on Earth, right, is the amount of energy.
Yeah.
So that's one way to define life. Well, the input is energy, and the output is what? I don't know. Self-replicating. Wait, okay, let's go there. How do you personally define life? Do you have a favorite definition that you try to sneak up on? Is it possible to define life on Earth?
I don't know. It depends on what you are defining it for. If you're defining it for finding different life forms, then it probably needs to have some quantification in it so that you can use it in whatever the mission that you're operating.
So you mean like it's not binary? It's this is like a 7 out of 10 on
lifelessness. I don't know. I don't think that defining is that essential. I think it's a good exercise, but I'm not sure if we need to agree a universally defined way of understanding life. Because the definition itself seems to be ever evolving anyway, right? We have the NASA's definition. It has its minuses and pluses, but it seems to be doing its job.
Well, what are the different? If there is a line and it's impossible or unproductive to define that line, nevertheless, we know it when we see it. Is one definition that the Supreme Court likes, and that's a kind of an important thing to think about when we look at life on other planets. So, how do we try to identify if a thing is living when we go to Mars, when we go to the different moons in our solar system, when we go outside our solar system to look for life on other planets? It's
unlikely to be a sort of a smoking gun event, right? It's not going to be, hey, I found this.
You don't think so?
I don't think so. Unless you find an elephant on some exoplanet, then I can say, yeah, there's life here.
No, but isn't there a dynamic nature to the thing? Like it moves. It has a membrane that looks like there's stuff inside.
It doesn't need to move, right? I mean, like look at plants. I mean, they grow, but there are plants that can be also pretty dormant. And arguably they are they do everything that one of my favorite professors once said, that a plant does everything that a giraffe does without moving. So movement is
Very
not
Zen statement.
necessarily.
But on a certain time scale, the plant does move. It just moves slower.
Yes,
It moves pretty.
I would say that
quickly on a time
it's hard to quantify this or even measure it, but it is a life is definitely chemistry finding solutions, right? So it is chemistry exploring itself and maintaining this exploration for billions of years.
So a planet is a bunch of chemistry, and then you run it and say, all right, figure out what cool stuff you can come up with. That's essentially what life is. Given a chemistry, what is the cool stuff I can come up with?
If that chemistry or the solutions that it embarks upon are maintained in a form of memory, right? So you don't just need to have the exploring chemical space, but you need to also maintain a memory of some of those solutions for over long periods of time. So that's the memory component makes it more living to me, because chemistry can always sample, right? So chemistry is chemistry, but are you just constantly sampling, or are you building on your former solutions and then maintaining a memory of those solutions over billions of years? Or at least that's what happened here.
Chemistry can't build life if it's always living in the moment. The physicists will be very upset with you. Okay. So memory could be a fundamental
life is not just
life.
Life is obviously chemistry and physics leading to biology. So this is not a disciplinary problem of one discipline trumping other discipline. It's that, but what you need to have is definitely, chemistry is everywhere, right? I tend to think you can be a chemist, you can study chemistry, you can study physics, you can study geology anywhere in the universe, but this is the only place you can study biology. This is the only place to be a biologist.
Earth.
That's it.
Yeah.
So definitely something very fundamental happened here, and you cannot take biology out of the equation. If you want to understand how that vast chemistry space, how that general sequence space got narrowed down to what is available or what is used by life, you need to understand the rules of selection, and that's when evolution and biology comes into play.
So the rules of natural selection operate to you on the level of biology?
Rules? I don't know if there are any rules like that. It would be fascinating to find in terms of the biology's rules. That's a very interesting and it's a very fascinating area of study now, and probably we will hear more about that in the decades to come. But if you want to go from the broad to specific, you need to understand the rules of selection, and that is going to come from understanding biology, yes.
Well, actually, let me ask you about selection. You have a paper on evolutionary stalling where you describe that evolution is not good at multitasking, or like in populations that evolve quickly. I mean, it's a very specific thing, but there could be a generalizable fundamental thing to this, that evolution is not able to improve multiple modules simultaneously. I guess the question is, what part of the organism does evolution, quote unquote, focus on to improve?
Yeah, that was the driving question. We meddled with the part where you shouldn't be messing up with translation. This is the...
Should or should not?
You shouldn't. As I said, there are many ways to break it, and all life needs it.
That's one of the your favorite things to do is to break life to see what happens.
It's because that's how kids learn, right? So you have to break something and then see how it will. Then you do it over and over again to see if it will fix itself in the same ways.
Yeah.
So it's our, I don't know, it's the most fundamental properties of ourselves as human beings. So if we shouldn't break translation, then we should try to break it.
Yes.
To see how it will repair.
So which part did you break?
I broke elongation. So
what's the role of elongation in this process?
So we have 4 steps of the translation: to initiate, elongate. So to elongate the chain of the information chain that you're now creating, the peptide chain, or let's say broadly polymer chain. And there's the termination step and there's the recycling. So all of these steps are carried out by proteins that are also named after these steps. Initiation is the initiation factor protein. Elongation is the elongator protein. We broke elongation. So the starting codon could still arrive to where it's supposed to go, but the following information couldn't get carried out because we replaced elongation with its own ancestral version. So we inserted roughly a 700 million year old elongation factor protein after removing the modern gene. So we made this ancient modern hybrid organism.
And that essentially creates, in some way, the ancient version of that organism.
I wouldn't say so. It's a hybrid organism. It's not necessarily because the rest of this cell, the rest of the genome is still modern. Mm-hmm. And that goes back to the difference between Jurassic Park. There are many differences, obviously, given that this is not fiction, we're doing it. But also, we are not necessarily... I think in Jurassic Park, they are taking an ancient... they find an ancient organism and then put a modern gene inside the ancient organism. Mm-hmm. In our case, we are still working with what we got, but putting an ancestral DNA inside the modern organism.
So you're like taking a new car and putting an old engine into it?
In a way, yeah. Yes.
And seeing what happens.
Yes, but in our case, it's more like a transformer than just a regular car. It is doing things.
So it's a more complicated organism than just a car.
Okay.
Yeah. I got it. So what does that teach you?
We wanted to understand multiple things. One is how does the cell respond to perturbation? And we didn't just put the ancient DNA. We inserted, we sampled DNA from currently existing organisms, so the cousins of this microbe, mm-hmm. And collected DNA sequences from the cousins as well. So both ancestor and the current cousin DNA, so to speak, and engineered all of these things to the modern bacteria and generated a collection of microbes that either have the ancient component or the variant elongator component that still alive today, but coming from a different part of the tree.
So you broke elongation. Was that something you did as part of the paper on evolutionary stalling to try to figure out how evolution figures out what to try to improve? Did that help?
Yes, because we were not supposed to mess with the translation. That's exactly what we did. Mm-hmm. And we altered elongation by changing it with different versions of elongation that are either coming from species that still are around today. You can imagine them as sitting on the tips of the tree near far branch, to compare to the organism that we're working with, distant cousins, as well as the ancestors of the bacteria that we are now modifying.
How much different variation is there in that elongation step? Like what are the different flavors of elongation?
That's a very good question. So mechanistically or mechanically, it's the same. It's very conserved. So all life elongates the same way. It's nothing but a shuttle. You just carry the chemical with you, the bit, to the heart of the machine.
Is that essentially doing like a copy-paste operation?
It has its tail that's attached to the code, which is then carried biochemically to the linear chain to the core of ribosome. And it sits on there. It releases, and the peptides click, the codes rather click. Once that chemistry that is at the tail end occurs, the protein leaves the center. So you can imagine it's like it hops in there and hops out. Mm-hmm. And when it hops and hops out, it leaves the information behind. That's all it does, is bring the information, get out of there. And it's all triggered by biophysics, biochemistry, because of the way the enzyme chews energy, in this case GTP, how the phosphor leaves the center, that gives the additional kick to the enzyme to leave the center.
So which parts are different then? Where's the flavors of different flavors of elongation?
Usually the parts that matter don't change over time. Nature conserves the sites of these proteins that are important for its job. If there's a difference, then we want to know, especially if there's a difference between two cousins, and we look at the sites that interact with the most important parts of this machinery. If we see any difference, we tend to mutate or we revert, we engineer that part, we alter that part because it gives us a clue that there must be something interesting going on here or not.
Okay, so that's not the fundamental part of the machinery, but it's some flavorful characteristic that you can play with.
So now you stripped the machinery down to its parts, and now you are looking at the parts of the part.
Okay.
And it depends where you're looking and how you're looking and what you're looking at. But usually we see up to 70% level conserved identity across all modern versions. When you travel back in time, the identity decreases. So elongation likely existed. We have good reason to think that it existed at the dawn of life. So you're looking at a 3.8 billion year old mechanism. And when we look at the ancestors that we resurrect, we see about 40% identity. So the identity definitely decreases as you go back in time, but still 60% shared information over 4 billion years is pretty good.
Is that just for elongation or for the entire translation?
Depends on what you do. So for initiation, we've also recently published this. It's a different story. But overall, you see high level of identity that is kept intact, especially if the component is essential for life.
Okay, so 40% and 60%, 70% you said, but like from generation to generation, how does evolution, and presumably that's what that paper is looking at, is the parts of the parts.
Yeah.
How does it able to say like mess with the parts and try to come up with a cooler, improved version of the organism?
Yeah, so let me describe to you what we did in that experiment. We took we took bacteria. We perturbed the elongation in all of these with different variants. So we had an initial set of a group of bacteria that we had. We then subjected these bacteria to evolution in the lab. All right. So we first of all we knew we broke it because upon engineering we measured what's going on with the cell. It's not growing as well. They're not healthy. We can see it with our eyes. We can measure it. That if they were generating an offspring every 20 minutes, now it is 40 minutes, right? So we really messed them up. They don't want to work with this thing. They don't want each other, but they need each other. So we created that situation for them, which is good because we want to see how we wanted to see how they will cooperate with each other to fix this problem. Because we know that that's not the condition that they want to live in, especially when they know what they can do. So with that, we subjected these organisms to evolution in the lab. That's we refer to this as experimental evolution. We subject bacteria to different selection pressure, project them through bottlenecks. Every day, we randomly collect a handful of bacteria from the flask, put them in a new fresh environment with fresh food, keep them in this environment for 24 hours until they reach a more dormant state, and then we introduce them to a new environment. So we repeated this for about, I will say, 150 days. So every day, not nonstop, we repeated this experiment.
Some kind of, how many different kinds of environments are there?
We kept the environments to the same, because we had different initial conditions. We kept the environment constant, same temperature, same food, same source of carbon, but we created replicates for each lineage. So in some ways, we created our own fossil record in the lab by evolving and generating these flasks. And every step of the way, we also froze these cells and took stocks of them in the cryo freezer.
How long does it take to go from one generation to the next with bacteria?
If you for E. coli, it's usually 20 minutes.
Okay, great. So that's the experiment.
That's the experiment.
And you're always messing with it in the same way for the initial.
It's the same
condition?
way. So we introduced variation at the elongation level because we perturbed it with different elongations. We found that if we introduce a different protein that is very different, the cells don't like that, right? So if the distance is larger, the consequence is also large, meaning that you hit them harder if you introduce a variant that is really foreign to them, that's really distant. In our case, it was the ancestor. They really did not like the ancestor, but they were okay with their nearest cousin.
Right. Okay, great. So you did vary in the distance.
We varied at the evolutionary distance, and then we kept the experimental conditions the same, and we propagated these populations every day for 150 days, and we collected bacteria at every step of the way and looked at the sequence. We wanted to understand what sort of changes may have happened in the genome to respond to the variation that we've introduced.
So what kind of changes would you be seeing depending on the evolutionary distance of the thing you shoved into it?
Exactly. So we knew where we punched, right? We punched right the heart, right? We punched the translation. So we expected, is it going to be, is it translation? Are we going to see a change? Will translation respond to this by fixing itself right away?
Mm-hmm.
Or will it be another outside of translation, something completely different, a different module, because translation itself is a module? Or will it be within elongation, a really sub-protein level thing? So we had a strategy to identify the mutational pathways by categorizing what we expected to find or where.
Okay, so why does it not do multitasking? Why is it not improving multiple things at the same simultaneously?
It turned out that what we observed in general is that, first of all, the harder we hit the cells, the more likely they were to respond by changes right at where we hit it.
When you say hit it, you mean like change something about
I like to think of it as hitting because I like to think as breaking the cell, right? I mean, not breaking enough to kill it, but we still, because they're still alive, they're not doing their job well.
So the bigger the evolutionary distance of the thing you put in there, the harder the hit is how you think about it. The bigger the hammer,
exactly.
Do you hit it with? Okay,
that's what it turned out to be, because that's what the data told us, that if the variation is higher, then the consequences will also be higher, in the sense that the cells will not grow as healthy compared to a variant that is coming from a near or a variant that is coming from a near evolutionary distance.
Is it wrong to think of this kind of hitting as akin to a mutation, or no? What are we supposed to learn from this hitting? Like how the thing evolves after it's being hit in this way? What does that teach us?
Because we see translation machinery as almost it is so conserved and so essential. It is not even clear whether we can remove some of the parts or whether the entire translation will need all of the same parts in the same efficiency. Mm-hmm. We don't understand the rules of this machinery. So the first thing we want to understand is that what is the resilience? What are we really talking about here when we talk about you cannot mess with this translation? Is this true? Because it is so conserved and so similar and functions in the most conserved ways, that was the first thing that we wanted to understand.
Did you learn anything interesting about the resilience at the chemical, physical, informatic, computational?
No, I wouldn't say that. I think in the biological level, yes, because we found that the different modules started responding to the changes that we've introduced, and that we could never recover the translation as effectively as it used to be. So that it never reached to its optimality, that it was always suboptimal. It needed, say, one more mutation perhaps to get there. It accumulated 4 mutations. We did a lot of experiments to understand this, of course. It was accumulating mutations. It was getting better at its task. Maybe it needed a couple other mutations to get really good at it, but somehow those mutations never happened. And before those mutations happened, we saw another module emerging through mutations and getting better at its own different task that is not translation. You can think of cell as a web of networks, right? And we think of these as multiple, almost airports. There are proteins that are more central hubs versus there are proteins that maybe are not as important hub. If you introduce a problem in the most populated hub, you're going to mess up the traffic system more drastically. And that's what we were messing with in the biological terms as well.
So when we say module, like translation would be one of the modules.
Translation would be one.
So you're basically saying when you mess with translation, the organism would choose to either try to fix that module or another module, depending.
Exactly.
But it wouldn't do multiple modules.
It wouldn't do multiple modules. It focused on one module at a time. And right before that module maybe reached to its own maximum, it stalled its optimality at a certain degree. So you never get to a degree that is more optimal than you can achieve, even though perhaps another mutation could get you there.
Since you messed with the translation, from a sort of optimal perspective, wouldn't it make sense for the cell to try to start fixing the translation?
That's exactly what we thought, and it was not the case for all the broken translation machineries. For instance, if the variant was coming from a near ancestor, that didn't happen. It was almost cruising around, trying different modules and sort of living its best life still, without because there's no real urgency in the system to fix the most important problem.
And there's
perhaps.
also not a direction. Maybe to you it's obvious that's the problem, but to the cell, maybe you're the problem. I'm living, like you said, my best life. Like we don't. I mean, I guess that's the thing about evolution is we don't know what the right direction to.
Yeah, it's almost like you can imagine that you have this messy closet, and you
Go on. Happens to be an accurate representation of my life.
So
but
you take a look at it and you see all these sweaters or jeans or all over the place, and then you look at a drawer that has socks coming out of it, and you think that's the most important one. I'm just going to fix that one, and then you fix that one, and then you think you will get to the other one, but you don't because you just fix the most important one. That is the whatever that was getting into your way. That's really what evolution is. It's quite lazy. It fixes the problem that seems to be the most immediate, and it doesn't go beyond what it really needs to. It seems like, at least for our experimental setup, that was the case.
Especially for rapidly evolving systems. So, is the environment they're operating in pretty constrained? Like, is there urgency?
I would say that we definitely constrain the environment. It's definitely removed from their natural setup. We are not evolving them in a gut. It's a very homogeneous system, very controlled temperature, controlled food, controlled carbon.
So just looking at that, let me ask the romantic question. How did evolution create so much beautiful, complex variety on Earth? Like from that, you're saying that we're talking about improving different modules, but if we step back and look at the entirety of the tree, of the different organisms it created all throughout history, the stuff that's fun to you with the first few billion, and the stuff that's fun to me when I watch on YouTube, which is like the lion versus gorilla fights and so on. But the whole thing is fun. So with all that beautiful variety from the predator and the prey, from the self-replicating bacteria and all that kind of stuff, how did it do it?
How is a very difficult question, especially when we don't understand the past with clarity at all. I can tell you that there seems to be very critical innovations that happened throughout the history of life that are each themselves very sophisticated singularities that emerged once and then they set the tone. One of which is emergence of translation. It seems like it happened once. It had to happen once. Seems like that's all it took. 3.8 billion year, maybe older. Clearly subjected to a lot of chemical evolution even prior to the last universal common ancestor. And then you jump and you see emergence of cyanobacteria that undeniably changed the course of this planet in the subsequent aerobic photosynthesis that life learned how to utilize what's available in the environment in the most profound way. And then you move forward, you see the emergence of eukaryotes, the endosymbiosis, also another singular event. And then you move forward, and then comes the plants. So these are, I counted, I think, 6 different things that seems to have happened just once. And
the singularity events in the history of evolution of life on Earth.
So what's really fascinating here is that there seems to be 2 different courses, the time course. Evolution is operating at the molecular level, right? We're talking about seconds. We're talking about mutations that happen every second. We're talking about selection that's also happening under a minute, right? So that is a very fast process. The fact that I can evolve bacteria in a lab, and I say almost complainingly, "Oh my goodness, it took me 150 days." I mean, that's pretty rapid for a change to be seen. But then the big changes and the ones that I'm talking, the really big innovations that caused an increase of oxygen on this planet, or even its own mere presence, are due to these molecular innovations. Seems to only happen a handful of times over billions of years of time scale.
Let me ask you this question having to do with my half asleep tweet. So, saying that we all originated from one common ancestor, that's just one of the miraculous things about life on Earth. Of course, you could say there's multiple common ancestors in the beginning, multiple organisms, and so on. But the other stuff that you're talking about is these singular events, these leaps of invention throughout evolutionary history. Now, there's a bunch of people who were commenting, a bit surprising to me, who were basically skeptical of this idea.
The idea of,
Well, I would say evolution, honestly, the process of evolution. But when you just actually focus in on like, holy crap, eukaryotes were invented. Holy crap, photosynthesis was invented. Like, those are incredible inventions. And also, we can even go to Homo sapiens, like intelligence. Like, where did that come from? There's these mysteries. I think where that skeptical comments were coming from were also just the general skepticism of science. I think from the pandemic, people, maybe a failure of institutions and so on, there's been a growing distrust of science. And it's not so much that it's anti-evolution, it's more of a stepping back and saying, wait a minute, maybe scientists don't have it all figured out. And I think to steelman that case is almost a step back and to realize there's so much mystery to each of these leaps. So it makes you wonder, is there something that in 100, 200 years we'll figure out that we totally don't understand yet? Like I talked to a bunch of people about another mystery, which is consciousness, right? And there's people called panpsychists who believe consciousness is one of the fundamental laws of the universe. So there could be, like we have laws of physics, there could be something that's like a consciousness field or something that permeates all matter. And so like there might be, it's kind of like Newtonian physics versus general relativity. Like we have a good understanding of how things happen, but we need another layer of understanding to fill in the gaps of the mysteries of it all. And that sort of is a sobering reality that maybe there is something we really deeply don't understand. Do you have a sense of where the biggest mysteries here are? Is it at the origin of life itself? Is it the leaps that we're talking about? So you see the beauty, you're fascinated about the translation mechanism. What are the deep mysteries there to you?
we are nothing but chemical systems capable of formulating or answering questions about our own existence. Right.
We humans or all of life, you think? Oh,
Humans. No. Humans are. I mean, the fact that we even have this conversation about our place in the universe is, at least to our knowledge, is quite specific to our own chemical species. But
yeah, it's kind of wild. We're introspecting on our evolutionary history, and we're just a couple of organisms.
Yes, and
with like another organism listening to this, and like their mind blown. There's like 3 organisms, 2 of them talking, and the third one's like, "Holy shit!"
I think that understanding what I really find interesting about understanding origin of life, or even contemplating about our own place in the universe, if at the end of this would come down to appreciating, or even before appreciating, really truly comprehending what it is that we got here. That to me is a huge gain, because there's no single question in biology, I think, that will give that that will deliver that magnitude of that message and understanding, but understanding how life here started at first place. If we truly comprehend that, this is not a concept that is well thought in schools. We ask students to memorize these concepts. If they are lucky, they learned RNA world, chicken and egg problem, etc. That's the extent to which that got. Maybe their biology teacher was personally interested in the subject matter, if they're lucky. You know the saying that the uh, brains are evenly distributed uh, across any metric you can imagine, but opportunities are not. So if people aren't understanding the importance of this, is because that's a lack of opportunity right there. That was skipped through the proper education and training in the delivery of why science matters or how science actually
Yeah, but
works.
how do you even begin to seriously think about the origin of life? I mean, every problem of existence, of life, has its time. So I don't know if it's time to understand consciousness yet. We might be 100 years away from that. The origin of life, I don't know if it's time for us to understand that yet. Maybe we need to solve so many more problems along the way. And
It's
so,
not a competition of problems, right? So there are all kinds of problems, and it takes a lot of people to make the world. So you will always have some interesting brain going after an interesting problem to their own. The issue here is that we need to first of all understand that what we have going on this planet is pretty good. Good planets are hard to find. If we are
yeah,
alone in the universe, that's huge. We need to take care of what we got here, and we are incredibly vulnerable to the changes that our own species also helped create on at the biosphere, at the ecosystem level. We take it for granted. We take what we created for granted because of the fact that we think we are some sort of ultimate endpoint, the most sophisticated, amazing thing that nature could generate. I think
yeah,
understanding, and not even us understanding, but asking these questions of where did this even come from? How did this even begin? And attempting to understand that using chemistry and physics and biology, and because we can, that's the ultimate gift we can give back to the entire species on this planet.
yeah. I mean, it's humbling. It's humbling to realize the complexity of this whole mechanism. It certainly puts humans in their proper perspective. That we're not just because we have brains and brains are intelligent doesn't mean we're the most intelligent thing. Because ultimately, the whole mechanism of nature seems to be orders of magnitude more intelligent. All of it, like we're like a hierarchy of organisms that have a history of several billion years, and that all somehow came together to make a human. And there'll be life after us, just as was life before us. And something that comes after will be perhaps even more fascinating.
Yeah,
Uh,
I think when you understand the magnitude of what happened here, there is no room for arrogance. It should overwhelm you and humiliate. It's pretty humiliating.
yeah.
It's quite amazing what happened here, and there is no other discipline that will deliver that but exploring our own origins and looking at life as a more planetary system phenomena rather than one single species at a time, a collective look.
You mentioned this question in your TED Talk is the two possibilities of the universe being full of life and the universe being empty and we're the only life in the universe. Um, how do you feel about both options? Just actually, you as a single chemical organism introspecting about its existence in this world.
It's having a planet full of life is interesting because we talked about life being all about chemistry exploring solutions,
Mhm.
and having solutions in front of you is great. It's beneficial, right?
solutions being different organisms, like other humans. You see them as a solution to a chemistry problem.
Different. Yeah.
Oh, that's an interesting solution. That's not next time we're in Austin, so there's a bunch of weirdos. Every time I see a weirdo, I'll be like, "Oh, that's an interesting solution to this chemistry problem."
So now you think like an origin of life science, but
it's funny that one worked out. Let's see where else it goes.
But having this emptiness and the unpredictability of uncovering a novel solution can also have its own benefits, and we should be open to what other solutions might be out there and exploring those solutions.
Oh, to different chemistry problems. So that's what you see. You see the other planets out there as different chemistry problems.
To their own local environment, yes.
So how many chemistry problems have solutions that are lifelike to you out there in the universe?
It's a wide open palette, if you think about it. I don't quite know. We know the chemistry is chemistry. I don't think the chemistry will be different elsewhere. But again, what is selected by chemistry will be determined by the environment, most likely.
See, I think there's life everywhere out there. So there's a guy named Nick Lane whose gut, and it's interesting to me, I wonder what you think about it, is his gut is there's life everywhere out there, but it stops at like the bacteria stage. So he says that eukaryotes is like the biggest invention and the hardest one.
I wonder if he thinks that's an accidental outcome. If he thinks that's inevitable, I wonder what that means. But it's a likely possibility that the bacterial or microbial life is definitely more attainable.
So that's a weird world where our entire galaxy just has bacteria everywhere.
So if you don't like microbes, you are on the wrong planet.
No, I know. Yeah, and viruses. I don't know which one there's more of, but they're both, and most of them are like productive.
They're fascinating. They do everything for us.
If you don't like microbes, you're on the wrong planet. You're full of good lines. Okay, right. I just can't. There's like an imperative to the whole thing. To me, the origin is the hard question. But once it gets going, I just don't see.
Go ahead.
It seems like it's constantly creating more intelligent things, more fascinating, complex things. They're able to solve. That's a very
interesting. I definitely agree that the initial steps may be the ultimate determinants. That once you cannot stop it once it starts. It's possible, right? And
I just have never unearthed. Maybe. I just. Whenever I see life, it seems to flourish everywhere. The thing is, the only thing I haven't seen is the start of it.
Exactly. But and how are we going to understand that if we don't do origin of life science? I mean, that's the question here isn't exactly our ability to recapitulate everything that happened in the exact way that it happened, right? This is about what can happen rather than, or maybe how.
You think it's possible to study the origin of language using English? So, like, there's a very particular chemistry here. There's a particular set of assumptions, understanding about what life is, what everything is. Our perception of reality is very specifically constructed through the evolutionary process. I wonder if it's possible to get to some first principles, deep understanding of how life originates in such a way that you can actually construct it on other planets. Ultimately, it feels like if you're doing it in the lab on Earth, you're always going to be using some aspect of the life that's already here.
So that's what I sort of talked about in my talk as well. And
everyone should go watch the TED Talks. Very good. The annoying thing to me about TED Talks, I guess that's by design, is they're too short. It's like, come on.
And did you know that there's no prompter involved?
There's no wait, there is or
there isn't.
Yeah, you have to memorize stuff. It's
And
It must be a
thanks to my
grueling process.
amazing editor who probably is watching this too, David Bielo, that
Yeah.
it was very, very helpful. But I
Very
would say that
professional organization. I like this podcast. It's a very professional organization. I respect that medium. Yeah. Anyway, in the TED talk about life creating life.
so there's a likely scenario that once we understand how life as a chemical system is capable of formulating its own expression and generating a memory and manages its existence on a planetary body for billions of years. Once we understand what conditions gave rise to that, we may be very likely to understand whether a different planet also be likely to instigate its own chemical revolution if it was provided by through some missing ingredients. You can think of it as a sending fertilizer to a different planet that is missing its own chemical composition or lacking, or that it needs more of what it has. The difference between making that planet Earth-like, this is not what that's about. We're not talking about terraforming, or we're not talking about turning that planet into Earth-like system. We are talking about first understanding that planet, studying its chemistry, studying its properties well enough to understand whether it is close to its own chemical revolution, and maybe giving it that extra nudge. So this is obviously a pretty big speculation and suggestion, and it's a very interesting proposition because this is a yes or no question, right? This is the ultimate would you rather. And I think it says a lot about the perception of the person who's answering this question. That if the answer is no, absolutely not, that's not something we want to do, I want to know why that is the case.
So just to be clear, what we're talking about is looking at the chemical cocktail of a particular planet. Yes. And having tasting it and seeing what's missing. So having a very systematic, rigorous scientific process of understanding what is missing. Not what is missing in terms of to make it Earth-like, but what is missing in order to be sufficiently have the spark or the capacity of the spark to launch the revolution, the evolutionary process.
Exactly.
And then the question is, do we want to then complete the cocktail?
The proposition is to also make us think that we will likely have this capacity at some point, especially when we understand origin of life better and better, right? So we will be asking ourselves this question. I guess I wanted to bring this to daylight a little bit because maybe in 10, 20 years, maybe more.
So you wanted to ask the ethical question: should we basically start life elsewhere on another planet,
or enable the chemical capacity of that planet that it may one day itself get there?
Okay, so for me, the answer is yes. So if you were to try to argue against my yes, what would you say? Why not? What's the worst that can happen if we seed another planet with life? What are the things we should think about? Is your main concern a chemical biological one, or is it an ethical one? What do you think about?
Well, the worst thing that can happen is that it wouldn't work, right? So that it's not likely. It's not likely that any attempt like this would work. That's probably because how do you?
So?
You gotta be very. You have to have an understanding that I don't think we have just yet.
I see, because if it doesn't work, then we could try again, right? To me, the worst case, the thing I would be worried about is we create life. I mean, the same stuff I worry about, like with plants, is things that might have a conscious experience. And then the dark aspect of life is increasingly complex life. Maybe I'm anthropomorphizing, but it seems to have the capacity to suffer.
Huh.
And so we're creating something It's like when you have children, you put creatures into this world that will suffer, can suffer, and may suffer, depending on how you view life, may likely suffer. And so now you carry this responsibility for doing your best to alleviate any suffering they might go through. And that perhaps that's romanticizing this notion of life. Perhaps bacteria are not capable of suffering, but perhaps it'll create more complex life forms that would be able to suffer. And that feels like a responsibility as well. Of course, other people would be concerned. The more obvious concern is like, well, you just created a life form. How do you know it's not going to be a super deadly virus that somehow is able to hurt humans? Yeah, my concern is more, I feel like that's a solvable problem. The problem of creating conscious beings that are able to suffer, that's a tricky one.
Yeah, I can see why. Because it goes back to, again, would we, first of all, do we have a responsibility to propagate more of this chemistry that we have on this planet elsewhere, given that we know ultimately we will be vanished? By we, I mean the entire planet. And if this is in fact a very rare chemical event that happened because all the right circumstances came together and we were the lucky one, do we have a responsibility to sponsor it? This is if we were to back up
sponsor, I like it. That's a good way to put it. Yeah.
If we try to back up remnants of our civilization, right? So we wanna potentially create conditions on the different planets so that humans can survive, given that we know or we want to, just for the sake of growing.
Yeah, propagating, becoming multiplanetary species.
Exactly. But what really is at stake here, I think, is actually, or what is really more interesting is what we don't see, which is the chemical behavior that enabled everything at first place. That's different than sending potato crops or engineering bacteria to live on a different planet. That's very different. You are really stripping it down to what is possible at the chemical level. So even if you are instigating the chemistry on different planets, you are letting that very planet to do its thing. Mm-hmm. You're not necessarily contaminating this planet with different chemistry, because the idea behind this, at least the way I thought about, is that you understand that planet, you understand the conditions, you understand the chemistry of the planet really well before choosing the planet as a candidate at first place. And then it's not about sending a missing ingredient per se, but again, just sending more of what it already has. That would be respecting that planet's condition too. So I'm not suggesting any occupation. I'm not suggesting any colonization. I'm not suggesting any, let's just strip everything and make everything Earth-like. That's not what I'm saying. It's more about empowering that place. What you are saying is likely to be the motivator behind all this. That's not, because I see suffering, I see pain. It's very interesting. I think this is a question that really reveals a lot about the person who's answering it.
Well, okay, so the pushback on my pushback, if I so am deeply troubled by suffering, then I should be probably paralyzed about the history of life on Earth. And, there's...
Can you elaborate? What do you mean?
Most of life who's ever lived suffered in ways that are almost unimaginable to me.
You mean our own species?
Our own species and before. And animals living today. And we're not even talking about factory farms. We're just animals living in extreme poverty in the jungle. People think like in a natural environment, animals live in a happy place. No, it's a brutal place of desperately trying to survive, of desperately trying to look for food.
Yeah.
And it's just like all of that life, that's just mammals. And we understand mammals, but like throughout like trillions of organisms that led up to those mammals, and the organisms living everywhere, like even bacteria. There's death everywhere. So maybe this idea of death, this idea of suffering, is actually this thing that we see as a bug is actually a feature.
I don't think suffering is a linear property like that with life. And I may be with Nick Lane on this one, that the likeliness of anything similar to what we got here evolving in another planetary body, I think is quite low.
Where would you say is the biggest unlikely thing? Do you mean humans, or do you mean even multicellular organisms?
Probably multicellularity. It's but I understand the both sides of the equation, right? In one level, I can see that we may not have any other choice but to back up this chemistry somewhere else.
Yeah.
So you would be saving. It's the ultimate saving, or the record our own record. It's not about, yes, let's also save Beatles and all the amazing songs, but this would be the ultimate repository of life. But I can also see your point of view, for sure.
It's really interesting. So, like, don't see the plant with a missing ingredient. Try to understand what the ingredients it has. Is it possible to construct life? For me, from a computer person, it just feels like something that could be solved computationally.
We can learn from the mistakes that we've done here and aspire not to repeat them. It is possible. We do amazing things as humans. There's a lot of suffering, but there's also a lot of beauty. And we could choose what we want to be, or what we want to see, right? So these attempts need not to come from a place of fear, but it can come from a piece of hope and love.
I think we're just very recently figuring out stuff, like we've even just a century ago, we're doing atrocities that weren't seen as atrocities at the time. I mean, I think we're learning very quickly of what is right and wrong.
Yes. And I work with a lot of, maybe because I'm at the university, I get to teach young people every day. Even at the time of 4 year or few years, you see generational difference already unfolding in front of you. And maybe that's why I see hope, because I think what we get to interact with in classrooms every year is just getting better. They are aware of issues in a way that I sure wasn't at their age. In some levels I was, but in many levels I didn't think about. I wasn't concerned of the problems. Well, they maybe have to be concerned because it's hitting the reality, is hitting them hard. But younger people are not afraid of these things. An 18-year-old can face these brutal facts about the planet in ways that I don't think any other generation before them did.
Yeah, it's super cool. And like there's all these cool technologies that aid in the process of a human being being able to see the truth at deeper levels. Like, Wikipedia and just the internet in general is enabling education at a level that was unimaginable before the internet.
Yes. And I think space exploration, even contemplating about these possibilities, ultimately, and I will emphasize this again, should make us think about our own place in the universe. If we are alone, that is quite fascinating. And we definitely have a responsibility to guard what we got better and protect it better and don't take it for granted. If we are not the only one, that's also a lot of responsibility to understand what else is out there. So either proposition, as famously being told, is fascinating. But as a scientist, I think, and I think that's the general behavior, maybe not, my fellow scientists listening to this can correct me if they aren't like this, but you need to have a level of optimism and hope. That's something, that things are worth working for, worth dreaming, worth imagining. And we cannot just have fear of suffering or fear of pain stopping us from doing marvelous things.
I've talked to quite a few people in my life who've gotten a lot of shit done, have helped a lot of people, and I don't know a single one of them who's not an optimist. Now, there's a place for critics and cynicism in this world, but in terms of actually building things and creating things in this world that help a lot of people, I think optimism is a requirement, is a precondition in almost all cases, in my limited, humble human experience. But I tend to, when I look out there, think that aliens are everywhere. I think to me, I have a humility about, I tend to see us humans as being very limited cognitively. Like there's so many things we don't understand. I think eventually we'll understand. Of course, we don't know this, but my gut says we'll understand that alien signals and life has been all around us, and we're too dumb to see it. Like whatever life is, whatever the life force is, whatever consciousness is, whatever intelligence, whatever the mechanism that led to the origin of life on Earth was everywhere, and we were just too dumb to see it. It's in the physics. It's somewhere. We'll find it somewhere in the physics.
I think that's one of the most humbling parts of also being a scientist, that we know that we never know for sure. And for the outsiders, perhaps, that may be a very um, strange way of living, especially when your pursue is about creating knowledge, and that you'll know that what you created can also be, and hopefully will be, disproven, so that another level will rise. Um, and I think we've seen that this lack of maybe connection between the approach to science or knowledge versus folks who are maybe not thinking about these problems every day, that we are okay with being wrong. That's, in fact, we know that that's the only way to push the limits of knowledge.
How do you think life originated on Earth? We've talked about this a bit. Do you have a gut feeling about? First of all, actually, even to step back, do you think, because you were like flirting with this idea, did the translation mechanism came before life?
I think that you cannot separate from translation, emergence of translation machinery from emergence of life, or something like translation machinery. This whole informatic chemical computing system that is also capable of dynamism and evolvability that comes with biological behavior from emergence of life itself. We definitely took a lot of steps towards understanding origins. We are able to create molecules from right environments, lightning, heat, and you make amino acids. So we are able to create the building blocks, the Miller-Urey experiment. That's now like 60 years ago. We are able to create the building blocks. We are able to make them interact with one another. They can get more complex. Some call this messy. There's all this chemistry that's going on. We are able to have these chemicals interact with one another, maybe um, have even some emergent properties that we can quantify. Definitely there is this trend towards more systems level approach to origins, with more introduction of systems level chemistry, more network level chemistry, and complex system integration, in order to understand how, now that we can make these building blocks, we can make them interact with one another, but how do we make them interact with one another in more intelligent ways that will have the properties of a biological system, will be heritable, it will be responding to the environment, it will mutate, and it will sustain itself. That is the final bit, I think, in our origin of life adventure, and we are extremely close. I'm very optimistic that our community will get a handle of this problem in this decade. This is, in fact, I think, one of the most exciting times to be doing this work.
What would be super convincing to you, like incredibly amazing, would blow your mind if it was done, X was done in a lab? Like what would you, I mean, I don't know if you would call it origin of life, but something really truly remarkable and special done in the lab. What would that look like to you?
This properties that I listed, this was 5 properties that I listed about, and machinery that is capable of sensing and responding to the environment. If we can I would imagine it's similar to Miller-Urey experiment, where they only sparked in particular environmental forces and were able to produce a chemical that is important for life, or a mix of chemicals important for life, or building blocks rather. I would, if I saw that a similar experiment where a well-defined geochemical parameter was subjected on a mix of chemistry, which led that chemistry to form some level of computation informatic biological property, and by biological I'm going to keep it to very minimum, um, as I defined early on, that would be super exciting to me. A self-organizing chemistry that we can create experimentally in a flask by simulating the conditions of early Earth, be it radiation, be it temperature, or mix of both. That would be very cool.
And doing all the five, the chemical, physical, informatic, computational, biology.
Yes.
So like simulation and a computer would not be good enough.
It would be great because they help to understand the parameters, maybe formulate, maybe quantify, create models, but ultimately you need to experiment.
Unless it's quantum mechanical simulation, but that's going to be extremely difficult. So simulating from the physics up, that's going to be very, because you're going to have to simulate the physical, the chemical, the informatic. I mean, honestly, it's very difficult to start the quantum mechanics and end up in biology all through simulation.
Yep.
But the stuff that DeepMind did with AlphaFold and protein folding is really inspiring. It's inspiring in that you're able to solve a difficult biology problem.
Yeah, absolutely. That's why there's definitely a lot of benefit to those models, predictions, because they at least help the experimentalist to come up with the priors and parameterize things better, maybe eliminate very obvious dead ends early on, given that experiments take such a long time and it's a huge investment. And no one's a better experimentalist than nature, so...
Let me ask you perhaps a depressing, sad for you question.
You really want to make me sad. You're not going to win.
No, I know. There's a flame of optimism in you that will never be extinguished. Okay. The idea of panspermia. You mentioned, would we seed another planet with life? Is it possible that our planet was seeded with life from elsewhere?
So what the proposition I made, I like to think of it as protospermia rather than panspermia, because it's even more, it's a more proto state than the acknowledging, because in panspermia you still have a cell, right? You still have something that is very, even a cell to me would be very Earth-like.
Yeah.
Right. I'm talking at subcellular level in the proposition of spreading chemistry.
So spreading chemical ingredients, not spreading life.
Exactly. It will be more like the fertilizer that is well adapted and compatible with that planetary body. In panspermia, you're still imagining either an entire bacteria or microbe or a cell or something that is DNA, which is still Terran.
So in that sense, that doesn't matter to you because it's chemistry. That's the initial conditions. It doesn't matter how the initial conditions came to be. They are what they are, and let's go from there.
Yeah.
And there's all kinds of fascinatingly different initial conditions in terms of chemistry at different planets.
Yes, but in terms of panspermia, I mean, obviously there's going to be always room for those sort of discussions, or there will be those discussions will always be present, I think, in any life in universe debates. But the problem I have with panspermia is that it removes the problem from the planet to somewhere else. It makes it very difficult to answer scientifically, right? You just took the problem away from this planet and formulated in a way that I cannot go and try to understand in the lab doing experiments or even through models.
Does it though? So I've heard brilliant biologists like yourself say that, but I just, to me, okay, here's how I think of Earth. So I actually am able to hold all these possibilities in my head, and all of them are inspiring to me. I kind of think there's a possibility that Earth is just an experiment by a graduate student, by an alien graduate student. Like,
so I know the exact episode of Star Trek you're talking about.
but like there is some, to me, that's inspiring. If we are,
but that's not what panspermia is about. That you're talking about my proposition. That's not what panspermia is.
what's panspermia?
It is.
Life just came from elsewhere.
Mhm.
Still, that's interesting because there's still giant leaps that happened on Earth, it seems like, beyond the initial primitive organisms, like eukaryotes.
I don't think panspermia usually articulates at the level of eukaryotes. I think they talk about bacteria primarily. I think so.
Right. Mm-hmm. So that's still interesting because all the different leaps of evolution still happen here on Earth. That's still interesting.
Yeah, but it's definitely interesting to listen to, but I wouldn't place it in. I wouldn't know how to place it in the studies of origin of life, I guess, or early
Here's
life.
how we place it. You have the initial conditions for the origin of life, and you try to create life in that way that you've described in the 5 components, and it keeps failing. So what panspermia allows you to do is to also consider the question, maybe there's missing components.
How do you answer that question?
Through exploration and through science.
Yes, yeah.
Looking outside of Earth, looking at the fundamentals of chemistry and physics.
How do you understand that with fundamentals of chemistry and physics?
How do you understand gravity?
But you're talking about panspermia, right? Just I don't understand how would you. It's different than if you think it's similar to looking for life in the universe. Is that what you're thinking?
No, I'm saying there's a missing component that came from elsewhere.
But a whole entire organism is not a missing component like that, right? I mean, when you're thinking about origin of life.
No. That's an assumption. Your assumption is all the ingredients for the origin of life are here on Earth. Now, I tend to believe that most likely that's the case. I'm just saying it's inspiring to think that there is some ingredients. You're going to push back because that's not panspermia. That's pros. But
see, okay, so think. But yeah,
it's also kind of fun to push back on you. No, I understand. I understand if actually a living organism ended up here from elsewhere, that means a lot of the exploration we're doing here with the ingredients that we know will not give us the clues to the origin of life. But it just seems like it's still very useful to try to create life here, and then we'll see, wait a minute, don't you think we'll be able to show that panspermia is very likely? Like if we just keep failing, we understand biology deeply, understand chemistry deeply.
I don't think so. I mean, there will be the failure is not going to indicate that this must have been. I don't think anyone will put the problem to something else just because our experiments failed.
So failure means we don't understand the chemistry deeply enough.
Yeah, and we, given the progress we made and how many brilliant people are working on this right now, and it's definitely more. I would say that we are approaching this problem in more broader ways, with different ways possible. I'm confident that we will get there. For us, again, we are interested in early cells and first cells and what followed the origin of life, but we cannot, given that it's a continuum that's between the origin and emergence of first cells, it's hard to separate these two ends from one another.
So given that life is a solution to a chemistry problem, if we reran Earth 1 million times, how different would the results be? If we look at that wheel, how different would be the tree of life, do you think? Like, what's your gut say?
My mind asks, are you imagining, if we're repeating the planet 1 million times, are we seeing, are the things that happened, I'm not talking at the chemical level, but at the environment level, do they happen at the same time, at the same frequency, at the same intensity every time you're running this tape over and over again?
Yes. You mean like geological stuff?
Yeah, like is the
So
same.
you're saying those are important. I mean, that's
Yep.
the fact that you would ask that question is also fascinating. So that's important. The timing, the frequency, the intensity of geological events.
So when we run this imaginary rewind and replay experiments in our minds, I want to know whether we are positioning all the same geologic events at the same chronological order as well, or whether we are also giving them more randomness. So if the volcano erupted, is it happening at the same time? If you have are dinosaurs getting wiped off every time with the same meteorite that's hitting the same?
But also like temperature changes and all that
changes
kind of stuff.
Everything.
That's actually, I've heard you say somewhere that one of the things that's fascinating to you about this whole process of evolution is that the process of evolution, all the mechanisms were invented and developed despite all the variation geologically through the hardship that Earth has gone through.
That the biological innovations persisted despite that.
Yeah, despite that, which is interesting. You kind of think of the biological innovations kind of happening on their own.
Because we actually have a center exploring this problem. We want to understand whether it's almost like judging a book by its cover, right? Do you just look at an environment and then see whatever is present or scarce in that environment and then think that, okay, the life form that will exist in this environment will obviously have a lot of molybdenum in its system. Look at all this molybdenum around here. Mm-hmm. Or will it be because if you say that, you are now putting the environment in the more prime driver role, right? That you're saying that environment will determine what biology will or will not use. But we've done studies that show that it's not necessarily this straightforward. That, for instance, we looked at going back to nitrogen. One thing that's fascinating about the way cells fix nitrogen, the ones that can do, is that they also do this through a lot of help of a lot of metals, a lot of elemental support, really. And which geologists use to understand where did this metabolism even evolve, where at first place. So we look at ancient oceans, we try to understand the elemental composition of ancient oceans. And what we see is that in some cases, the metabolisms, even though they prefer a certain metal or an element that is in the environment, that metal wasn't abundant in the environment, but still life chose that. So it's not that straightforward as though whatever you are what you eat, but you don't necessarily eat what is obvious to you. Just because there's a lot of that food around you doesn't mean life will ultimately go there. Maybe most of the time it will. But it seems like in the case of nitrogen fixation, it didn't, and maybe that made the difference.
It's so cool that, right, it's not the abundant resource that's going to be the definition of what kind of life flourishes. So it's not a straightforward thing.
Yes.
But your sense is that the different timing of the different conditions of the environment would change the way evolution happens.
Yeah, for instance, I mean, I think it's in the 80s, maybe earlier than that, the Stephen Jay Gould's book, Wonderful Life, which changed, I think, a lot of scientists' life, including mine. He contemplates on this notion of the tape of life. Of course, I hope people still know what tape is, but I think your listeners will know what tape is. I don't know. It's the
tape. Go on, tell me about the tape. Tape. Is that like a TikTok? Can you swipe on it? I'm sorry. Go ahead. I apologize for my rude interruptions.
I kind of asked for it, but he speculated or suggested this hypothetical experiment whether if life was recorded on or can be imagined to be recorded on a linear chain of events recorded on a tape, and if we were to rewind this tape, would we listen to the same song, right? So this was, and in his proposition, I also thought, yeah, but are we replaying the tape in the same exact manner, or are we, meaning all the geological and environmental events, are they happening at the same time? Because then you removed the randomness from equation a little bit, right? You just removed it because you're assuming everything will happen at the same time, at the same intensity. So that's not too contingent. That means that the natural selection you're thinking is really operating at more, or evolution is operating at more under more random forces. then that can be dictated by the environment. So in our way of understanding or thinking about rewinding, replaying, I don't think we're thinking about the role of the environment as clearly, or don't seem to be integrated as much.
But I also wonder if it's possible that the chemistry ultimately defines the destination. That despite all the environmental changes, despite all the randomness, we'll end up in something.
But we are not talking about whether life will emerge and sustain itself. We're talking about whether life will emerge and sustain itself in the shape and form that is similar to what we have right now. So you are chemistry, I'm chemistry. We're having this conversation, and your plants are chemistry too. They're also having their own conversation.
These plants are fake, but yes.
I knew that. I didn't want to say that, but they're fake. But
do you look at my place? Of course, they would be fake. Otherwise, they would die.
What's wrong with this place?
It's wonderful. I'm Alice, and this is Wonderland. This is great. This is great. It's just that this is a place where robots flourish, and those plants are fake.
Are you saying that you and I are the only living organisms? Well, obviously there are microbes in this room, but
yeah, we are the only living organisms. I was thinking of getting a dog.
Well, you don't. This is not a clean room, so you have microbes here.
Yes, many millions.
Yes, so you and I and all the microbes in this room, we are chemical systems that are operating in a way that we can respond and sense and our environments and whatnot.
Yeah.
But if you are asking if we're going to be here, then you're imagining that another solution is also possible, which is different than the fundamentals of life, because life will do its life thing.
Mm-hmm. I guess it goes all the way back to the things we were talking about, translation, and the stuff you were messing with is figuring out what is the important stuff and what isn't. It makes me wonder about, just like with the translation machinery with human beings, I wonder what's the important stuff. Is it important to have two limbs? Is it important to have eyes? Like, it was obvious that the sensory mechanism of eyes, like sight, were to develop. How many times, if we were on Earth, would the sensory mechanism of sight develop? And what would it look like? Would it be one giant eye? Or would it be two? What's with the symmetry? Why are we so damn symmetrical?
In response to Steve Jay Gould's proposition,
yes.
Most people who argue that life is convergent and it will in fact lead to a few determined outcomes, or it's not that the outcome is determined per se, but it's the pathways are restricted and the mutational trajectories that life can act upon are already very limited, so that the final outcomes are a few, and eyes being one of them. So the convergence at the eye level was presented as an example of why life may actually embark on the same solution over and over again, given that many species evolved it independently from one another.
Do you think there's any inkling of truth to that? Like, is it just us humans thinking we're special?
I think those innovations came again so far after the I know it's the fun stuff. Yes, because it's.
Thank you. I mean, thank you. I think we humans tend to talk about the later stuff, but without the earlier stuff.
So when we think of early Earth, and I ask this to my students too. I want them to close their eyes and think about just nothingness but dust. We don't have trees, we don't have plants. When we say an empty place, or visually at least, we are talking about a planet that is really alien. So understanding our own past is similar to understanding an alien planet altogether, given that it is a very different planet that did not have any oxygen for 2 billion years. There's nothing that is familiar to us that we would even think about when we think about life that is present in our past, yet here we are.
So cool that from that came this, like houses and people.
And we are very, very... We are the super late arrivals to the party, right? So this is definitely not our planet. It's the microbial planet that we live in.
But the potential to create us was always there.
Like how do you know that?
Because we were created. Oh, I don't know. What is it? You think it's possible that even for the early stuff, yeah, maybe if it's super unlikely, yeah, that we just got this is the planet that got really lucky given the chemistry. Like maybe to create the bacteria is not so lucky, but to create complex organisms all the way up to mammals, that's super lucky.
Yes, and it
Maybe.
may all come down to a few innovations that happened at the molecular level that may or may not be inevitable. Uh, so all these molecular tricks may have enabled the sort of mere existence of whatever you are able to define as familiar to yourself.
And you have a hope that science can answer these questions to reconstruct.
Science is answering these questions. I mean, we are limited to going back to the beginning in our ways, right? So we rely on biology. It is overwritten. You're talking about 4 billion-year-old records that is ever changing. That again makes it beautiful, but also makes it difficult. It's not tractable. Geology has to some degree, it has a record of a more static, frozen state record that is embedded on itself on the surface of this planet, if we can find them. And that's the key, that most of these recorded remnants are, if we're lucky, we find them. They are not naturally selected. They're found. They need to be found for us to read them. So we work with a very handful set of samples, especially when we talk about the deep past, the planet with no oxygen, when we pass the great oxidation event threshold, that is about 2.5 billion years. So the earliest life is even harder. You are trying to write the story of life based on a handful of rocks and what is recorded on them.
Speaking of finding select remnants of our deep past, you said that you've been thinking about Nick Neelan's essay on scientific knowledge and scientific abstraction. So let me ask you, where do you think scientific questions and answers, or in general ideas, come from? You're a scientist. You ask very good questions and try systematically and rigorously to answer them through experiment. Where do you think ideas come from?
So ideas come all the time, right? There are all kinds of ideas. There are good ideas. There are not so good ideas. There are really exciting ideas, maybe some boring ones. But if you are really interested in doing something different, then you need to be willing to take the risk to be wrong. And that's incredibly difficult, even though we talked about the idealist sort of notion behind science that we ultimately want to be rejected, or our ideas need to be rejected for the entire infrastructure to move forward. There is a level of risk taking, I think, behind any creative idea, and I mean that in a true sense. If you are disappointed that your idea didn't work, then it wasn't a risk, because you still hoped that it will work. Mm-hmm. True risk is that you accept that it may not work, so that the failure shouldn't also surprise you.
Yeah. Is it when you embark on stuff, when you embark on an idea, do you actually contemplate and accept failure? Like as a
consciously,
possible...
I wouldn't say so, but I eliminated a lot of the things out of my work line by simply not feeling like studying them. I was bored chasing certain questions, and
So you trusted the signal of boredom as a good sign that it's not a good question.
It should definitely be, whatever you're doing should be exciting to you. If there's only one person that should be ever excited about what you're doing, that should be you.
Yeah.
And that's enough for that idea to go somewhere. I think that you need to believe in the idea, but at the same time, I think it's important to not fall in love with your mistakes. You know, that if something isn't working, you should let it go instead of trying to fix it, even though you feel that this is a mistake, or you know that it's a mistake. In order to instead of trying to fix it, you should wrap it up and move on to something else, which is incredibly hard.
Good advice for science, but also good advice for relationships. But okay, so but that's actually really hard, especially, I mean, this is like PhD stuff. Like if you sink in so much of your time, not even PhD, the entire scientific careers, it's really tough to let go.
Yes, and there is not a lot of room for true freedom. Maybe at this certain degree. So first, you need to be trained, right? It's not that scientists are just brilliant, amazing humans. I mean, they just know and learn how they know how to do science because they're trained in how to do science. So that is important because as someone who wants to definitely, I'm hoping that I'm giving the message that this is for everybody, that there's this notion of scientists being super smart people. That's definitely not true, right? It is a method that you learn to solve a problem. That's really what science is. And some are really good at it, and they get better at it under really good guidance, maybe good mentorship. And ultimately, everyone finds their own style of problem solving and what sort of problems they solve. But I have not met a scientist that finds their own pursuit boring.
Well, it can happen, but they're not going to be effective, just like you said. I think it's kind of interesting because in the age of social media and attention economies and stuff like that, I've interacted with a lot of folks like YouTubers and so on. I think a lot of their work is driven by what others find exciting. And I think that ultimately leads to a life that's not fulfilling.
I can see the reason behind it, or perhaps there's fear of failure that can be a major determinant of that pattern, like, right? So you try to do something that is accepted by others because that's maybe likely to give a result right away. But it's a long game. It's a very long game. And if you are aiming a long-term change and long-term impact, you got to be very, very patient about it. And you better tame your ego.
I mean, on YouTube and those kinds of places, on the internet, on social media, you get feedback like right away. And so it's even harder to be patient.
Yes.
So because the change in ideas develop over a period of months and years, if not decades, and the response from social media and so on is on the rate of seconds, minutes, and hours.
So I recommend actual physical libraries for people who may want to appreciate or remember the sense of time and how long it takes to build something. I think it is. You're right that the immediacy and the right response of and the fact that these places are, the algorithm wants you to respond right away and interact with itself, right? So I can see the appeal, but true innovation, I think, it doesn't even scream. It's not shiny, especially in the beginning. But it's also important to not fool ourselves and think that everything that people criticize has some super important meaning behind it. So it's a mix of the technique, the methods, and your gut feeling.
Yeah, and a weird dance between learning and accepting the ideas of the current science, and at times trusting your gut and rejecting those. Because science progresses by sometimes rejecting the ideas of the past, or sometimes building on them in a way that changes them, transforms them.
Yes, and I think what is hard is to really drill down into a concept, right? So you can create a new thing, and then it may be appealing and gain a lot of traction, but to sustain that, to continue that, you really need to show the true expertise. And so it's not only about defining a problem, but then really systematically solve that problem, maybe over the course of decades.
You mentioned the library. I've also saw that you've translated scientific documents, or at least like mentioned that you did it at some point in your life. So let me ask you, how much do you think is lost in translation in science and in life? How many languages do you speak?
Two.
how much is lost in translation in science and in life between those two?
But it's actually three because science is like another language, right?
It is. I speak Russian, a little bit of French, and it's always fascinating to see how much is lost. And the Soviet Union has a tradition of science and mathematics and so on. And it's interesting that a lot of the wisdom gained from that part of the world is lost, basically because of it was never translated.
Well, I'm not sure if it is lost per se. I mean, maybe it's more like a gain in some sense, right? Because you understand, and science is ultimately a human pursuit. So you cannot separate as much as maybe it's the best system that humans ever came up with to seek knowledge, to generate and make sense of the world. It works most of the time. It doesn't mean it's perfect.
Did the kind of translation you do, by the way, was for scientific work?
I directly translated for scientific work. Yes, I think that again, brains are equally distributed, but not opportunities are not, right? So if you want to include, if you want to benefit from all human power, whatever we can generate as human beings, you need to include everybody on the table, and that is by extending the opportunity. Is I think most of us that make it tend to think that we did because of something special about ourselves, but it is important to know that no, we were given opportunities, and that's why we are here, not because there was something inherently special about us, or that the system truly selects for the ones that really are.
Yeah, and language is a part of the opportunity.
Yeah, language is
Like
an
if you don't
opportunity because
speak,
comes with similar to bacteria, right? They speak these languages.
yes.
They have we call culturing the bacteria. We call it culturing, right? When we grow bacteria that we isolate from the environment in the lab, meaning that you create an environment for them to grow and thrive and sustain themselves. That's what we say. But culture is for microbiologists. For language, with language comes a different culture, a different perspective, and you bring that to the table. I mean, it brings the sense of diversity that can only be achieved by clashing perhaps two different cultures, two different languages, two different approaches, maybe in some cases four different approaches.
Yeah, I think language is not just a mechanism of communication. It's a way to, it's a dynamic system of exploring ideas. And it's interesting to see that different languages explores ideas differently.
Yes, and I think that so when I said science is like a language itself, I said it in two different ways. One is very, in a very literal meaning that you can speak English, but that doesn't mean you will understand a scientific paper. That it's a different level of English that you need to learn to understand. Even not just for scientific papers, even from discipline to discipline. I challenge any chemist to read an evolutionary biology paper, and vice versa. It may sound extremely different, a different language altogether.
Yeah.
But there's also the language of communicating, and because words matter, how we talk matter, how we represent our science matters. So yes, just learning English as a second language alone is not going to make you fluent in science either.
And it's interesting because in that sense, you speak many more than three languages because you're pretty cross-disciplinary. It seems like you have a foot in a lot of disciplines. I mean, you
Yes,
mentioned geology, biology, evolutionary biology. I mean, there's chemistry,
biochemistry,
bio,
biophysics, and
biophysics.
even we do a lot of statistics. So there's a lot of mathematics to what we do as well. Yes, we like to think of it as this. NASA's astrobiology program says it. I repeat it because it's fun. That it is not a fruit salad, but it's a smoothie. That's what we are generating.
It's not a fruit salad. So a smoothie uh, is a successful combination of those fields, and a fruit salad is not.
I wouldn't say it's success in necessarily. It's some if you put the wrong ingredient and you press the blender and you made it a smoothie. I mean, it can ruin the entire
Can it?
mix.
Can it though? Because I feel like
Yes, I can definitely assess that for ginger, for instance, that ruins every smoothie. I don't like
ginger.
it. I think so, but it's just a personal thing, and also I don't like cinnamon. But
The ginger has a cinnamony taste. Because I thought ginger was.
no, I don't think they do. But I also don't like.
Wasn't
So
that a
there are
thing
two
they add in
ingredients.
a lot of smoothies? I was forced a smoothie. I went to Malibu with a good friend of mine, Dan Reynolds, and he forced me to consume a smoothie. And it's probably the first smoothie I've ever had. Because I always was very judgmental of the kind of places and people that drink smoothies, but it was good. It was good.
Well, smoothie is very American, so
it is an American thing.
I wouldn't say success per se, but it is true that when you dance at the edge of different disciplines, that's when inevitably the innovation will rise, because you will see things maybe a little differently when you're on the edge, right? So, but it will probably take longer, and it may not be understood right away. It may not come into final form quickly, given that it is a new concept rising. So therefore, the patience will make more sense. Patience will be even more important. So if you are, in other words, if you are into immediate appreciation, that's probably not the way to go.
You're one chemical organism. So let me ask maybe a little bit more of a personal thing. Where did your life form originate? And what fond memory do you have from the early days of childhood that are representative of your bacteria culture?
I was born in Istanbul. So I grew up in Turkey. It's a city that has two continents, which is quite interesting. You have you see a welcome to Europe sign and then welcome to Asia sign the same day, depending on which part of the bridge you are. So that's where I was born, and I spent about roughly 20 years of my life, and then I immigrated to United States.
And it's a very proud culture. It's a beautiful culture. It's a very flavorful culture. What aspects of it is part of who you are? What are the beautiful aspects that you carry with you in your heart?
I think we are very sincerely human as a culture. I think that we have the saying that don't go to bed full if your neighbor is hungry. So, you wouldn't eat any food in front of someone where I come from without offering to share the bite. So I think those things, however small they may sound, a really big deal, especially when you are put in or move to a place that may not have those attributes. So I think that culturally there we had a lot of conscious, and just raw, deep human.
The connection. You value the connection between human beings.
I think so, yeah. I think I definitely carried that with me.
We talk a lot about biology. Let me ask you about the romantic question. What role does love play in the human condition, or in the entirety of life on Earth?
It's not easy to learn how to love if you're not loved. Okay? So it's something, but the good news is that it is something that you can learn. I think that's you can practice and teach yourself how to maybe give yourself the thing that wasn't given to you, and then ultimately give it to others. I think it would be quite arrogant to think that we will be capable of loving. It could be anything, really.
So just like translation, it's a repeating and a dynamical process
that you can learn.
Yeah, that you can learn.
Yes, and you should learn.
Yeah.
We should there's no excuse to not learning.
To love.
Yeah.
Because that's a deeply human thing.
It is a deeply human thing. It is a very sad thing if any one of us passes this planet without knowing what love is. And that could be a love to a pet, a love to a plant.
To a robot. Just kidding. Or a fake plant. We can't help who we love. What advice would you give to a young person today? High school, college? How to have a career they can be proud of, or how to have a life they can be proud of. You said an interesting thing about brains being distributed evenly, but opportunities not. It's really interesting to think about. I've talked to folks from Africa. You realize that there's whole areas of this earth that have so much brilliance, but unfortunately so little opportunity. And one of the exciting things about the 21st century is more and more opportunities are created, and so the brilliance is unlocked in all those different places. And so all these young people now have the opportunity to like do something to change the world.
I had a chance to visit Bosnia, so I was invited to give a talk in a very up north, like very northernmost part of the country that was impacted by the war tremendously, and it was a public talk. It was open to everybody in the village, and I was told even people drew from Sarajevo to attend. Whenever I think about our role as a scientist or the beneficiaries of the knowledge that we create, I always think about that night. That's how many people were in that room. It was incredibly crowded, and lots of young people who were trying to start everything new and not do or not carry replace whatever maybe the feeling that was taken from them with hope and love. Start a new beginning, be the seed for the next generation. And it moved me so much that they all came to hear about early life, space, something maybe different for them that maybe they were always interested in and never thought about. But what stayed with me was just the look and the look on their faces and the feeling in the room. The energy just was very moving to me.
Their willingness to be the seed, the first of their family and generation to do that big new thing.
Yes, and I
Take
will,
the leap.
and that's exactly why I'm telling this whole story, because for most of us, we may have to be that seed in our families, that the first one to do something new, to break that cycle, whatever it is that you want to break free from. I would want the young people to know that you can be that. That there are uh, just wonderful things to learn from this life, and it's just incredible to be living. And I would want them to know that their voice matters and they need to use it, especially those who think that their voice doesn't matter. And ultimately, I think what it comes down to is to trusting yourself, trusting and respecting your voice. If you're not loved, learn how to love. If you're not respected, start by respecting yourself. Learn how to respect yourself. You can teach yourself things.
Yeah, it's really difficult when you're surrounded by people that don't believe in you.
Yes,
So I think
definitely know the feeling, and I would just want them to know that they don't need to be defined by or reduced down to what others see in them.
believe in yourself, have the respect for yourself, try to develop the respect and the love for yourself. And then from that, it flourishes. You'll find others that'll give you love.
It may not. I mean, life is not fair. It's true.
Yeah.
It's be prepared that it's not very fair, unfortunately. And so I don't want to depict this Disney story and then yes, and everything will be just fine.
Yeah.
It's mostly isn't, but you learn a way. Life does it all the time.
Speaking of which, what do you think is the meaning of all this? What's the meaning of life? Why are we here?
Why we are here? Oof.
All the beauty you've discussed. Why is the translational mechanism machinery here? Why?
I don't think
Why so much beauty?
Why so much beauty? It is because we choose to see it that way. It's beautiful, but there is no meaning. I don't think. No. Yeah.
But why is it so beautiful? Why did we choose? Why From where is the imperative to see to see so much beauty in a thing that, scientifically speaking, or from a rational perspective, is void of beauty? It just is.
Not
Why
everybody chooses to see the beauty.
hate is gonna hate? I mean, we have the capacity to see the
We
beauty.
have the capacity, so why not use it to the fullest, right? We have the capacity.
But that capacity, isn't that fascinating? That we developed that? It feels like that was always laid in there in the whole process of life. This ability to find to introspect ourselves.
I mean, it's definitely soothing to think like that, but I don't think there is a meaning like that way. That's it's fascinating that we can understand it.
But why is it soothing? There's a desire, there's a longing.
But soothing doesn't mean that there's a meaning. Why is soothing a meaning? Let me just put it this way: because there is just, I think, so much unfairness going on. I wouldn't even dare myself to think that there's a meaning to out of respect to the ones that are suffering.
I see. I think out of suffering emerges flourishing and beauty. I mean, that's what I see.
I agree with you.
When I went to Ukraine and saw the people suffering, in their eyes and in their stories is a hope for the future, is a love for the people who are still living, is a love for life. So it's there. And that's the dark thing, is the suffering and the loss somehow intensifies your appreciation of the life that is still left. That's a weird thing too.
I think that there is something about still doing your best and believing that whatever the goodness is worth working for, is beyond, and to do that without a meaning, there's something more humbling and profound about that. And we have a this will come out very random. Okay, so just in Turkish bathrooms, there is this sign that says, "Leave it as you want to find it."
Yeah,
and I think that's a pretty good.
That's your meaning of life found in the Turkish. There's wisdom to that.
There's wisdom to that, but it also it's because however you leave defines you, right? So I think there's some profound meaning to that too. That just leave it as you would want to find it.
So that your little scribble in the long story of life on Earth is one that ultimately did a pretty good job. You know, it at least kept it the same as you found it.
or at least I left it in the way that I wish I found it.
Yeah, right. Oh man.
Yes, that's a wisdom from Turkish bathrooms.
That's where I searched for wisdom as well. And it's as we started with the origin of life and ended with the wisdom in a Turkish bathroom. I think that's a perfect conversation. You're an incredible person. The humor, the humanity, but also the brilliance of your work. I really appreciate that you would talk with me today. This was really fun.
Thanks for having me.
Thanks for listening to this conversation with Betül Kaçar. To support this podcast, please check out our sponsors in the description. And now, let me leave you with one of my favorite quotes from Robert Frost. In 3 words that can sum up everything I've ever learned about life: it goes on. Thank you for listening. I hope to see you next time.